want to reiterate how proud I am of all the students this year. All of you did amazing work under under any circumstances, but especially under the circumstances that we're in right now. It would have been very easy, I think, for all of you to easily justify not doing anything this summer, but you didn't. You decided to take on this heavy task of a remote internship, never meeting your mentor, and you've amazed me of what you have been able to do. So before we start, just congratulations to all of you. I know many of you are probably nervous right now, but you don't need to be. You're going to do wonderful. I'm so happy to be connected to you. So thanks for all that you did. Likewise to the mentors, again, just your willingness to give of yourself during these times, these uncertain times, lab shutting down. It's remarkable. I commend you for doing that. I know the students have really enjoyed working with you and have learned a lot from you. So thank you so much for allowing this program to continue. We're not 80 students this year, we're 20-ish students, so we're much smaller. And we're not in person today celebrating on this last day, which is always the favorite day. But you guys are here and you did work throughout the summer. So thanks to both of those groups, and to the Cancer Center for supporting us, the university, to the NIH for allowing us to be flexible, and the Doris Duke with our money as well. So I appreciate all of these things. I will have many more people to thank at the end, but I wanted to thank some people at the beginning, and mostly to congratulate you students. You guys were amazing. So thank you for all of your hard work. And I really look forward to these final talks. So a little bit about logistics of how today will work. I have up the schedule that everyone should have received. This one Zoom link will remain open throughout the entire day from 9 a.m. until we finish. However, we will have breaks. We have 4 different sessions. They will start on time. Students within those sessions, we're not exactly sure what time you will go, but we have the 9 o'clock session. We have 4 presentations. They will happen in those order. The way that we hope this will happen is that the mentors will unmute, introduce the students. The students will then share their screen, give a talk, 8 to 12 minutes-ish. Afterwards, we'll take a few minutes for questions where all the mentors can already unmute and ask questions. Anyone else who is here, please just raise your hand in the participant or ask the question in the group chat. Group chat is open throughout, so you can even ask your questions asynchronously, and at the end, the students can address them there. If there's an abnormal amount of questions, I'll ask to switch to chatting specifically so we can stay on time, but I don't envision staying on time being hard today. We've only put 4 to 5 talks per session, and each session is about an hour and a half. So once the session ends, all of the additional time until the next session begins will be a break. So we don't have to sit here in front of Zoom students for 9 straight hours. You're going to have time to get up and have a half hour between sessions to recover, get a drink, get some food, whatever you need. So that's kind of of how it will work today. And I think I'm just going to get out of the way and let you get started. So our first presentation will be from Jonathan, and I don't know if Matt or Giannis, if one of you want to introduce him. Let me stop sharing.
Thank you. Matt will introduce Jonathan.
Perfect.
Sure. I guess first I wanted to say thank you to everyone, especially Dr. Boone, who helped coordinate this program and meeting. My name is Matt, and I've had the pleasure of working with Jonathan on his mathematical modeling project in the Zervin-Tanakis lab. And today he'll be talking about his project, where he uses computational modeling to investigate drug resistance caused by the interaction of 2 cell types in breast cancer.
Thank you. I'll share screen now. You see my screen?
Yes, perfect.
Okay. So my presentation is titled Utilizing Mathematical Modeling to Elucidate the Differential Effects Fibroblasts Impart on Breast Cancer Sensitivity to Lapatinib. So to give a basic introduction to breast cancer, it's a major issue. 1 in 8 women will develop a disease in their lifetime. Approximately 20% of breast cancers are HER2 positive. So it means that on the actual cell, on the actual surface, of their breast cancer cells, they overexpress this HER2 receptor protein. So because they're overexpressing this HER2 receptor, there's more growth signaling being received by the cell. And as a result, the overall tumor is more aggressive. Luckily, there are HER2 targeted therapies which selectively attack these HER2 overexpressing breast cancer cells. But depending on the drug, approximately 38% to 75% of patients do not respond. So a critical issue, a current major vein of research is investigating what are the mechanisms of patient resistance to these targeted therapies. Stromal fibroblasts and lapatinib. So lapatinib is a critical HER2 targeted therapy. It's very commonly prescribed in the clinic, and it functions by nullifying the signaling being sent by that HER2 receptor to the cell to grow. Stromal fibroblasts, so these are an abundant cell type in the breast tumor microenvironment, and they have recently been demonstrated in vitro to mediate the lapatinib response. So if you culture some HER2 positive breast cancer cell lines with the stromal fibroblasts, the actual impact of lapatinib is severely reduced. So my project overview was split into 3 main steps. The first step being to construct an in silico breast cancer mathematical model in MATLAB. The model accounted for the protective signaling being sent by the fibroblasts, and it also accounted for the cascade in the PI3K ACT pathway. This cell signaling pathway is a critical one that mediates overall cell growth. The second major step of my research was actually analyzing the parameters of the model to investigate what are the fundamental drivers of this fibroblast protection from lapatinib. The last major step I took is actually simulating drug combination therapies to see what's the most effective means to arrest cell growth in these fibroblast protected cell lines. So I guess the main question in your mind would be why mathematical modeling? Why are we pursuing this specific vein of research? The main reason is that models are very flexible, so they can directly inform future lab experimentation and drug therapy design. So once you have a model that's been fitted to the data, it's a computational representation of a very complex biological system. So you can analyze the parameters of that model to gain a fundamental understanding of the system you're modeling. In this case, it's the HER2 breast cancer cell line under lapatinib assault and with the protection from the fibroblasts. Moreover, once you have the model, you can readily simulate drug combination therapy. So whereas it would be very time taxing, both costly and from manpower to simulate the cells under different drug combinations, by having a model, you can easily do this. And it's a very efficient means to help expedite research. So kind of shown by this graphic here, by having model predictions, you can directly inform future research, which will help you make better model predictions. This is the main construction of the model. So first I formulated the underlying mathematics, and then I fit the model to 2 different data sources: reverse phase protein array, RPPA data, and then general cellular viability data. So RPPA data is very neat because if you basically tear open the cells, you lyse them, and you run this array, you can gain relative levels of protein expression. So if you collect RPPA data at different time points, you can see how the expression of proteins in the PI3K ACT pathway, how it changed over time. Moreover, once you have cellular viability data, you can combine the 2 to get both a pathway representation of the model. You can gain how the cells grew overall, how the overall cell grew over time. Once I fit the model to these 2 different data sources, I ran parameter sensitivity analysis to really figure out what was driving fibroblast protection from lapatinib. And the last major step I took was actually simulating combination drug therapies. The 2 cell lines I modeled were BT474 and HCC1954. So BT474 is a fibroblast protected cell line. These 2 graphics are cell viability curves, so it's how the cell grew over time with respect to these different concentrations of lapatinib. So once you had no fibroblast versus fibroblast co-culture, the impact of lapatinib was negated. Even at the highest concentration of 3 micromolar, you didn't see cell death indicated by a downward sloping curve. This contrasts to a fibroblast insensitive line. So whether you had no fibroblasts, so monoculture, or you had a fibroblast, so co-culture, the impact of lapatinib was the same. The reason why you see higher cell growth in this HCC cell line at any given concentration of lapatinib is because this cell line has a very strong downstream PI3K mutation. So in that critical pathway, there's a very strong mutation that helps send pro-growth signaling independent of the HER2 receptor. So that's why you see just more cell growth in HCC versus BT474. The general model topology, so I had 4 key model variants that I simulated. I had the BT474 cell line under both monoculture, so no fibroblasts, and under co-culture. and the same different setup for the HCC cell line, so monoculture and co-culture. The model accounts for the HER2 receptor activating into its pHER2 active form. The pHER2 helps activate the ACT protein into its active phospholact state. This phospholact protein helps activate the S6 protein into its phospho S6 variant. Phospho S6 is a very critical protein because it helps phosphorylate a variety of downstream transcription factors and helps promote overall cell growth. The model accounts for lapatinib buffering the phospho-HER2 formation, and it accounts for fibroblasts helping turn over S6 into phospho S6. Depending on the model I was simulating, I had to fit between 19 and 27 parameters, and I used MATLAB's least squares nonlinear optimization method to do so. To give you an example of what the model, or what the underlying mathematics of the model looked like, I used chemical kinetic rate laws to describe the protein interactions of the protein species in the model. And I tied the actual kinetic rate laws into ordinary differential equations. These equations describe how the rate of change of each protein species in the pathway over time. So by using these ODEs, you can actually numerically integrate them in MATLAB and gain solution curves. So to show you what the model looks like once it's been fully calibrated to data, and to show you visually what fibroblast protection looks like, here's the BT474 co-culture model under no drug, DMSO, and under lapatinib. So when you have no drug added, you see this very upregulation of HER2 expression indicated by this solid line blue curve. Note the axis break, so it jumps from 1.8 to 6.2. So you see this wild upregulation of HER2 without lapatinib. Once you have lapatinib added to the cells, you saw a significant downregulation of the HER2 expression. But once again, you had that fibroblast protection. So because you had this co-culture model, the actual cell growth curves, these green curves, remain about the same. This is what the model looks like once it's been calibrated, and it kind of serves as a visual representation of how these fibroblasts are protecting the cell, even though lapatinib is working effectively. So the 4 chemo variants I mentioned earlier, they yielded an average root mean squared error of 0.86. So in a root mean squared error of a value of 0 is like a perfect model. So considering it's only 0.86, I'm working with this very noisy biological data. It was the best root mean squared error I could achieve. And I ran parameter sensitivity analysis to figure out what was driving fibroblast protection. So when you have a multivariate function, such as overall cell growth, and it's dependent on many different variables, in this case, the parameters of my model, in order to run sensitivity analysis, you have to compute the partial derivatives of cell growth with respect to certain key parameters, which is what I did. So the 2 key parameters I chose to run the analysis on was the phospholact catalysis rate. So the rate at which phospholact is able to activate the S6 protein into its phospho S6 variant, and the phospho-HER2 catalysis. So the rate at which the phospho-HER2 protein is able to activate ACT into its phospholact variant. This is the monoculture cell line for BT474, and it's the co-culture cell line. So you see a dramatic jump in the cell sensitivity to these critical pro-growth signaling parameters. This contrasts to what was observed for the HCC cell line. So whether you had monoculture or co-culture, the actual sensitivity didn't differ by that much. You see a drop of around 4 for the phospholact, you see a jump of around 2 for the phospho-HER2. So the main conclusion from this analysis is that when you have a fibroblast protected cell line, such as BT474, you see an unbalanced scale from monoculture to co-culture. Once you add fibroblasts, the cell sensitivity to these critical pro-growth parameters increases dramatically. Whereas under HCC, so when a cell line is fibroblast insensitive, there's a balanced scale. You don't see this major shift of cell sensitivity to these critical pro-growth parameters. So now that I had really investigated what fibroblast protection looks like, I wanted to validate my model before I went around just simulating drug combination therapies. So Corcola et al. Reported that lapatinib and a downstream phospholact inhibitor, they yield a significant synergy in PI3K mutant cell lines. So I wanted to see, before I just went around simulating combination drug therapies, I wanted to see if I could reproduce these previously published results. So I simulated this drug combination for the HCC cell line and the BT474 cell line. The HCC cell line had that very strong PI3K mutation. So if my model is correct, I would expect to see a very strong synergistic response between the 2 drugs, which is what I did. So this heat map describes the temperature of the heat map is the predicted cellular variability at 4 to 8 hours. So values greater than 1 indicate cell growth, values under 1 indicate cell death. This top row is DMSO, so no drug, the lapatinib, the phospholact inhibitor, and then the combination therapy. This column over here is the HCC co-culture model. So whether you had DMSO, so no drug, or lapatinib and phospholact, you still saw cell growth. Whereas once you had the combination therapy, you saw cell death. So you see that very strong synergistic response. Whereas for the BT474 cell line, even at the combination therapy, you don't see cell death. So my model is actually reproducing these published results, and it's been validated against an external data source. Next, I wanted to simulate the combination therapies, which will arrest cell growth and the fibroblast protected BT474 cell line. So I simulated lapatinib and a phospholipid inhibitor. I simulated lapatinib and then a fibroblast signaling inhibitor. This inhibitor represents an inhibitor that blocks the protective fibroblast signaling. And then I simulated a triple drug combination of all three therapies. The temperature of the heat map is once again the predicted cellular viability at 48 hours. This first column is the lapatinib and the phospholipid inhibitor. So if you inhibit around 99% of the phospholipid signaling, you gain pretty much no cell growth. Whereas if you inhibit around 99% of the fibroblast signaling alone, you still see pretty significant cell growth. The interesting thing is that if you actually combine the therapy, so if you're able to inhibit between 50% and 75% of both the fibroblast signaling and the phospholipid signaling, you gain a similar impact to what's observed for signaling to inhibiting 99% of the phospholipid alone. So the main conclusion from this simulation is that the most effective means to arrest cell growth in a cell line similar to BT474, which is once again fibroblast protected and carries a very, very weak P53K mutation, is to use a triple drug combination of lapatinib and then the dual phospholipid and fibroblast signaling inhibitor. Whereas, as we knew from previously from the Coca-Cola paper, the most effective means to arrest cell death in a cell line similar to HCC 1954, which is fibroblast insensitive and carries that very strong P53K mutation, the best means is to use lapatinib and the phospholipid inhibitor. And this is my mentor acknowledgements. Thank you for your help over the summer. Without you, wouldn't have been possible to do this project.
All right, so what I would like to do, so great job, Jonathan. I am going to unmute everyone so that we can give him a proper round of applause if we could, please. All right. All right. Excellent work, Jonathan. Are there any questions? So I just muted everyone. So again, we're still working on this technology. Are there any questions for Jonathan? We have time for one or two, if anyone has one. And you can type in the chat, you can raise your hand in the participant box. If not, Jonathan, okay, Taylor asked the question. Jonathan, what was the most challenging aspect of your research?
The most challenging part by far was actually the data fitting. So the underlying math was pretty straightforward, but the actual fitting to data was a very time-consuming process. I would have to run the scripts for hours overnight sometimes. So while the math behind the model was fine, the actual fitting to the data was really hard.
Excellent. Any other questions? Jonathan, I know I asked you this the other day when you practiced, but I can't remember now. Is there a thought of how fibroblasts are giving this protective effect to lapatinib? Is it a soluble factor that they're secreting? Is it something that's caused by the physical interaction with integrin signaling or something else? Do you know much about that? And whenever you say fibroblast inhibitors, is there an inhibitor that can inhibit that signaling that you're modeling?
Yeah, so the signaling is done by secreted cytokines. So they secrete these factors into the microenvironment, which are received by the cell and encouraged to grow in the face of lapatinib assault. An actual direct fibroblast signaling inhibitor that doesn't exist, but you can negate the effects of the fibroblast by targeting the downstream pathways which are activated by the secreted factors. So instead of actually saying, oh, I'm gonna make a drug that will impact these 100 cytokines, I would just say I will make a drug that will inhibit the pathway that's activated by the cytokines downstream. So like these inhibitors, like mTOR inhibitors, they do exist in the clinic.
Excellent, cool. Great work, Jonathan. So I think we'll move on now. If there are any more questions for Jonathan, please feel free to continue to ask them in the chat. And our next speaker will be Sam. So David or Paul, if you want to introduce him. Oh, David, we can't hear you, unfortunately, or I can't hear you. I don't know if your mic's not working. You are unmuted.
About now?
Yes, perfect.
All right. So it's my pleasure to introduce Samuel Cho, who's a rising senior at Montgomery Bear High School in Maryland. He is actually planning to apply to Pitt in the fall for the combined medical program with the goal of becoming a radiologist, and also to explore computational techniques in radiology to improve the state of the art of that field. And to get his feet wet this summer, he is learning to apply computational techniques to improve drug discovery. As you'll see, he's done a fantastic job. Sam?
Hi, I guess I'll start. So can y'all see me now?
We see you, but we don't see anything shared yet, Sam.
Oh, oh, forgot to press share.
There it is. We see it
There
now.
it is.
Perfect.
So the project I'm presenting today is the AutoDocker Pharmacophore package, or app. And that is a molecule, because this is about drug discovery. Now, drug discovery is the process by which new drug candidates are found. Typically, this involves the process of screening, which is you take a bunch of candidates and you essentially just work until you can filter them down to just one. And then that one goes to bureaucratic stuff. But what we're focusing on is this area. So drug discovery now, mistakes were made. So just to mention, this is a very expensive and time-consuming process. It can take nearly $1 billion and years to even get to a stage where you can test things. So what scientists do now is they use structure data. So now we understand that drugs are ligands that bind to receptors. Ligands are small molecules that bind to a larger receptor, typically a protein, changing its behavior. So for example, this is aspirin. I had to take quite a few of these during the project. And this is the COX-2 protein. In its normal state, COX-2 produces chemicals that are inflammatory. So the way aspirin works is it'll bind to these two sites here. And that causes the COX-2 protein to change shape and function. And this actually produces anti-inflammatory molecules. So that's how aspirin works. But it's also one of the reasons why why this is really helpful, because now that we understand that drugs are just ligands that affect certain molecules, researchers can now, if they know what they want to target, they can use computers to do a lot of that work for them, such as they can narrow down the drug candidates by computing which ligands would work for the receptor. They can reduce the number of dead ends and failed candidates, which is where a lot of cost goes in the later stages. And overall, it cuts down on time and costs. So, so what my project focuses on has to do with pharmacophore modeling. So so just to get a few definitions out of the way, I'll be using the word feature and receptor a lot. So a feature is a region or part of a molecule that has chemical properties. And normally this is divided into six categories: hydrophobic, hydrogen acceptor and donor, positive and negative ion, and aromatic ring. So, and a pharmacophore is the set of features which are important. In other words, they're the features that are responsible for allowing the ligand to bind to the receptor and do an action. Now, with the definitions out of the way, let's take an example. So here's a molecule that, well, as Dr. Boone shows, most of you wouldn't even be here without this. We call it caffeine. And so what we do is we identify the features, So here's a hydrogen acceptor, hydrophobic area, and an aromatic ring. Now, these can just be calculated based on, because we know that certain arrangements of molecules have certain properties, by figuring out if a place on an molecule has this arrangement, we can say that, oh, it has this feature. So what this gives us in the end is a pharmacophore, this set of features. Now, what we can use this for is something called virtual screening, Pharmacophore-based virtual screening, which searches through a database to try to match other ligands that have the same pharmacophore. And this is real. And so what you can do is, this is caffeine, and we put into a screening program, and then you can get a bunch of molecules. This is just 4 out of a couple of hundred, I believe, that showed up, and they all share the features of caffeine. Now, we really like this in computational drug discovery because this is fast. Like, it can take, in like a few seconds, you can narrow down your search from like 10,000 molecules to like 5,000, a few hundred. But there are a few issues, mainly the fact that not all features are important. Some features don't interact with a particular receptor, so they shouldn't make a difference. And if you include them, they can cause problems. Such as here, this is one of the molecules that putting caffeine's pharmacophores generated. And again, it shares all of caffeine's features, but it also has a hydrogen acceptor here because there's an extra bit. Now, if we were to try to put this through the screening program, then what we want to see is caffeine, because we know that works, but we wouldn't get it because caffeine doesn't have this here. It's looking for a feature that doesn't matter. So this is one of the reasons why ligands are actually really important, because without them, you wouldn't really be able to know if a feature was important or not. So by having a known ligand, you can figure out that one's features and match things up. So the question we're trying to ask is, can we figure out the pharmacophores without the ligand using the receptors? I mean, what if we could just remove the ligand from the equation? Like, there's no need for it. Let me see. That if we were to, let me think. Okay, so what if this wasn't the case? What if we didn't need the ligand data? What if the only thing we needed was a receptor and it generate the pharmacophore for us? And what if we automated this whole process? So that way, the only thing you needed to do was put in a receptor, press run, and you get your results. And what if I stopped it with the rhetorical questions and just said, there's an app for that? Yes, the entire title was a brick joke. It's the AutoDocker PharmacoFind package. And what it does is it's a package of 2 programs that processes and analyzes a given receptor. In this case, here's an adenosine receptor. And it'll generate a collection of what it believes to be essential features or pharmacophores. So the first step is through AutoDocker. And what AutoDocker does is it automates a process called fragment docking, which it identifies and tries to predict all the positions and orientations that another molecule would probably be in to bind. So in this case, we use probe molecules, which are small molecules that have that exemplify a feature that we want, such as here. It's a little hard to see, so let me zoom in. So here, these AutoDocker does dock multiple probes at once, but for simplicity's sake, I'm sticking with just one. This is benzene. It's hydrophobic and an aromatic ring. So we can see it really likes this area. So this is probably an area that we want to keep track of. So what we do is then we put into Pharmacofind the docked information. And what Pharmacofind does is it'll cluster the locations of these probes based on how close they are. And these clusters now represent features, regions where we assume this property will is present. Now, this is one of the earlier runs, so you see a lot of these features. And what Pharmacofind also does is it applies a set of thresholds, limits to filter it out, which at the current moment there are 2, which is how many probes are in a cluster and the binding energy of the cluster on average. And by doing this, we get what we hope is a pharmacophore, the essential features. Now, if we want to quantify how well it does, what we can do is we can take what we know of the pharmacophore, like the actual thing. This is the real ligand in this case, and this is what has been shown in experiments to be the important step in hydrophobic. And we can put this and calculate what's called the Tanimoto coefficient, which allows us to see the overlap between these two. The closer it is to 1, the better it is. In this case, it's 0, because here we have a hydrophobic area here, and it's not here. So this is pretty bad. So what we want to do now is to try to optimize the Tanimoto value by varying the thresholds. So what we do is we ran AutoDock on 95 receptors from the directory of useful decoys enhanced. Now, before I continue, I'm just going to say I'm all for toilet humor, but my mentors and the wiki calls it dude rather than, well, you know. So I'm going to call it dude. And then what we do is I ran Pharmacofind on the docked files with varying thresholds, just every combination of those 2 thresholds between a certain range. And then I calculated the Tanimoto coefficient for the generated features and the known features. And then I try to find the thresholds and combine for each feature that maximized the feature Tanimoto, and then combine them into one set of thresholds. Now, a few hours after starting the program, what I'd got was this. This is what I call the coarse grain thresholds. This is just to get a general idea of the layout. So what's good is we see this region here. So we're going to zoom and enhance. Now, the program took 14 hours to run. And that includes 2 crashes, each of them about an hour through the process. But after all that, what I eventually got was this. So this is actually really good because we definitely see that there is some sort of cluster, like there's definitely an improvement here. So it's not just random. There's a sort of gradient. So then these become our thresholds for this test. So then the Tanimoto calculated for each of these are as shown. Now these are, if you notice, they're all pretty low. And as I mentioned before, we want Tanimoto to be high. But this is actually pretty good in terms of just a first run through, as well as we definitely now show that having thresholds for each feature type rather than in general does make a good difference. There is a pattern, or at least a range in which the thresholds do best at. And it does generate pharmacophores, just not as well as would be ideal. So next steps would be to find are these pharmacophores useful for finding molecules, and if not, why not, and how can we improve them? So, and we could probably add more thresholds and sets, and yeah. So that concludes my project, and I'd like to thank Paul Franker and Dr. David Coase and the Department of Computational and Systems Biology, and thanks to Dr. Boone, Solomon Lifshitz, the University of Pittsburgh, and the Hillman Cancer Center. And we don't have a lab logo, so this is just the lab photo with the dogs. And any questions?
Excellent job, Sam. I'm going to do the same thing. I'm going to unmute everyone so we can give them a round of applause.
Praise me.
Excellent. Good job. So any questions for Sam? We have time for one or two. So Sam, whenever I talk at schools or I talk to people about doing internships, and I often tell them that you need to just get in the lab and try research. You've now done this 2 years in a row. Can you tell me some of the similarities that you had between the two experiences, or I guess, was it less challenging doing it the second time, knowing full well that you were going to have 14-hour runs that were going to crash because you were used to crashing and code's not working in the previous year? You're unmuted, Sam. I'm sorry, you're muted.
Well, the funny thing is last year that didn't actually happen. So this was, I actually had never even made a heat map until now. So yeah, it was a bit of a shock. It was the first time I was like, wait, this isn't done in like 5 minutes.
Yeah, it can be challenging. So there's a couple questions in the chat, so I'll go on to those.
About the computational resource question.
Yeah, so I'll read it and then you can answer it. If you have more computational resources, do you think you can do this faster?
Maybe. I'm not so great with the actual hardware, but I think so.
Were you running this locally? Were you running it on a cluster?
Yeah, I was running it on a Mac that really has no business running programs on it.
Yeah.
Like it almost caught on fire multiple times in the process.
Perfect. So probably then.
Yeah, probably.
Jenna, who you know well from last year, your mentor last year, asked, what was your favorite part of this project?
So favorite part of this project was, I guess, the first stage of it when I was just downloading the different files needed for this. And I was like, cool, it makes a molecule. And favorite moment in this project was where I first revealed the name and the reason I made the name app to my mentors. That was my favorite moment. One of them hated it, the other one loved it.
Excellent. One more question. What would be an ideal Tanimoto coefficient that you hope to achieve?
The ideal would be 1, but I'd say 0.8 or higher would be really ideal. I mean, chances are it'll probably just get like 0.6 or up, though.
Yeah, and if you were doing this, actually to follow up on that question, because I know very little about this, if you were doing this on a known interaction between a protein and a ligand and just modeled that known interaction, is that where the Tanimoto coefficients would be?
Yeah, so Dude gives us both the receptor as well as a file of, basically they manually or computationally found what the important features were.
Excellent. All right, thanks, Sam. Really well done. If there are more questions for Sam, please feel free to keep asking in the chat. But now, and if you can stop sharing, Sam. Perfect. Now we will move on to our next presentation from Oscar. Sang-hoon, would you like to introduce him?
Yeah, I'm glad I could work with Oscar Heller and introduce him for his final presentation today. I can say Oscar is a very diligent student. We had a Zoom meeting at 8 a.m. in the morning, and Oscar studied himself a lot about breast cancer and using supercomputer. So Oscar is going to present his research detection of somatic mutations in clinical trial breast cancer patient data. Please welcome Oscar.
Thank you. I'll share my screen right now.
Perfect.
Okay, can everyone see my screen?
Yeah, that's perfect.
All right, cool. Let me just minimize the Zoom as well because it's, okay. Hi everyone, my name is Oscar Heller and my project was called Detecting Somatic Mutations in HER2 positive breast cancer patients to predict drug response. Just some background, I did analysis on HER2 positive breast cancer patients, which is just an overexpressed protein in 25% of breast cancers. Many like antibodies, including trastuzumab, reduce mortality in various stages, specifically in stage 1 to 2, 3 tumors by 37% when combined with adjuvant chemotherapy. However, 25% of these patients do experience recurrence within 10 years, which has motivated a lot of new HER2 targeting drugs like lapatinib, pertuzumab, and nibertinib that have been tested in combination with trastuzumab. So this right here is a paper and a clinical trial study called CALL-GB that my project tried to mirror. Right here is the actual CALL-GB trial. Basically, they just randomized HER2 positive breast cancer patients into 3 groups that had different combinations of treatment drugs. For example, group 1 has paclitaxel plus lapatinib, and these groups have different combinations. This is arm 3 of the group or of the CALL-GB trial, as you can see. However, it was closed early due to its increase in toxicity for the patients. So therefore, they did analysis on arms 1 and 2, which led to their hypothesis and primary objective, which was to determine if the pathologic complete response or the lack of signs of cancer tissues in the patients after treatment in HER2 positive breast cancer patients from paclitaxel with trastuzumab plus lapatinib, which is this arm right here, achieved greater PCR. And this table right here are their results. As you can see, the TH and the THL arm, the groups 1 and 2, were not statistically significant, indicated by the high p-value. But there were some very interesting findings from the study, including that subsets of their population, like ER negative and PR negative, were statistically significant. Similarly, by PAM50, the HER2 enriched group was also statistically significant. And this is the PCR rate, and you can see there are observable differences between these 2 groups. So this led them to believe that there are a lot of underlying things involved with breast cancer that we just don't know, which motivated them to do machine learning by detecting somatic mutations along with a number of other things. So that kind of leads to my project, which is basically detecting somatic mutations from CALL-GB clinical trial data for 1, to be able to detect from all of the patients in the CALL-GB data, because they only tested for, or they only did the training set. 2, accurate detection of mutations from both RNA-seq and DNA-seq data. And 3, the comparison of mutation frequency in patients' drug response, which basically just refers to whether or not they achieve PCR. This is more basic rationale for my project and how it was going to play out. So we used the analysis of integrated RNA and DNA data through the pipeline unsecure will result in improved somatic mutation detection, which will inevitably result in improved predictive algorithms for patient treatment. And the reason we decided to do RNA and DNA data integrated was because it's been proven to boost sensitivity on low purity tumors, which basically just means that it has more accurate detection, which led to the hypothesis that 1, somatic mutations will be successfully detected in CALL-GB clinical trial data,
and more specifically that the frequency of somatic mutations between those who achieved PCR and those who did not achieve PCR will be different. This is the basic workflow for how my project works. Basically, the unsecure pipeline requires 3 inputs: whole exome sequencing tumor BAM data, whole exome sequencing blood BAM data, and RNA-seq BAM data. So right here, this is the RNA-seq BAM that had to be aligned to a reference genome with map splice. It was sorted, indexed, which are just basically ways of formatting the files. And these had to be pipelined through CRC, which is it's either, or it can be interpreted as a supercomputer or rather a computer cluster that can analyze a ton of data. Same thing with the whole exome tumor BAM data and the whole exome sequencing blood BAM data all had to be sorted and indexed to be run through Unsecure. And once that was done, Unsecure produced an output that was very difficult to interpret. So then that had to be annotated and made able to be interpreted by ANOVAR. And lastly, the last step was a predictive algorithm, which is more of an extension of the project that I'll get into later. So right here, this is some of the Unsecure outputs that are difficult to interpret. This is subject 10, subject 102, and subject 106. And these all come from a database called dbGaP, which stores confidential information about the patients, because which is where the COLGB clinical trial data pulled all of their resources from. So as you can see here, they do give the chromosome number, but there's some important information that's lacking, like the ID, the quality, the filter, etc. So therefore, it had to be annotated through ANOVAR. And these were some of the patients that were analyzed through ANOVAR. The blue ones are ones that did not achieve PCR, indicated by the 0 here in this red highlighted box. And we also picked ones specifically from the training set, so that way we could validate the results with the COLGB paper. There were also, as I said before, arm 3 of the analysis was cut off early because it increased toxicity in patients. So these were only from arms 1 and 2. And this shows all of their information. So these are a few results between from running ANOVAR and being able to interpret it. Right here on the x-axis are the types of mutations, such as frame shift deletions, frame shift insertions, non-frame shift deletions, non-frame shift insertions, start loss, and stop loss mutations. Right here is the frequency between in the patient samples. This bar right here, the gold bar, are those who did not achieve PCR right here, and the gray bar being those who did achieve PCR. As you can see by the error bars, there's really not a huge substantial difference between them because they overlap and they look observably similar. Similarly, we analyzed the patients for TP53 and PIK3CA mutation frequency, and we found pretty much the same thing, that there wasn't huge substantial differences between them. Here on the x-axis, you can see those who did not achieve PCR, and here are those who did achieve PCR. There are some differences, but mainly the frequency is pretty similar across the board, leading to the conclusion that, one, there are not statistically significant differences in mutations for those who did not achieve PCR and those who did achieve PCR. Also, in the first graph, there was the first column of frame shift deletions, which highlights its importance in playing a role in PCR. There's also no statistically significant difference between TP53 and PIK3CA in those who achieved PCR and those who did not achieve PCR. And the reasons for this can be numerous. The biggest reason being that there was a small sample size, as we only ran ANOVAR on 5 patients who achieved PCR and 5 patients who did not achieve PCR. And that kind of plays into both the extensions and the limitations. The biggest limit was that we only analyzed 5 subjects from each, meaning that there were only 10 subjects interpreted by ANOVAR because of time constraints, and therefore, for future experimentations, we want to detect somatic mutations on all 265 subjects from the COLGB paper and do a comprehensive analysis to find the new mutations between those who achieved PCR and those who did not achieve PCR. We also want to apply somatic mutation data to the development of drug response prediction algorithm, which, as I said before, by integrating DNA and RNA data to detect somatic mutations will result in more accurate predictive algorithms that can be applied to general clinical settings. These are my acknowledgements. Many thanks to Sang-Hoon Lee and Dr. Jiao-Sung Wang for their unwavering support and vital mentorship during this program, as well as Dr. David Boone, Solomon Lifshitz, Mark Krotek, the Department of Biomedical Informatics, and the Hillman Cancer Center Academy. I'd also like to thank the NIH and the Hillman Yes program, as well as the University of Pittsburgh, Central Catholic High School, and the various students who offered support either through Discord or direct communication.
Excellent, great job, Oscar. We're going to unmute everyone again. If everyone can give him a round of applause. Good job. All right. Are there any questions? We have a couple minutes for questions. Oscar, I'm going to start with one as we're waiting to see if anyone else has questions. So I noticed you picked 5. Again, it's a pilot study due to the limitation of time. You picked 5 patients from arm 1, 5 patients who had complete response, 5 who didn't. But I noticed 4 of them from each group were from arm 1. 1 was from arm 2. Is there a reason why you did that? And then actually, maybe I'll let you answer that question first.
Yeah, sure. So the decision to do, well, the patients were basically randomized, and we only stressed the importance of 5 on achieved PCR, 5 did not achieve PCR, just so that way we can understand the differences between the mutations that are correlated between each. So the arm choices weren't that significant. The only specification we had was it couldn't be arm 3 because that was closed early because of toxicity for the patients.
Excellent. And then a follow-up, you mentioned in the trial they found an increased benefit if the patients were ER negative. So
Right.
Did you consider that when doing your pilot too? Especially, I asked because P53 mutations are more likely in ER negative patients. So if there was positive PCR response, there may also be an increase in P53 mutations in that as well. So I was just curious if you thought about ER and PR mutations when doing your quick pilot.
I didn't stress the importance of that. I guess it would be more of an extension that I would do during the fall or the summer.
Yeah. Excellent. And again, of course, if you do all of them, then it's less critical. It's just when doing some selection. And let's see, we have a question from the chat. Asked, did you look at grouping mutations on pathways?
No, on the ANOVAR interpretation, the only significant mutations that were found were ones that involved frame shifts or ones that were stop-loss, stop-start. So no, we did not look at grouping mutations.
Okay. Any last questions? If not, again, feel free to continue asking Oscar in the chat. Well done, Oscar. And we're going to move on to our last speaker of this group, who is actually a returning undergraduate student. So Yaser has been here. This is his 3rd year. But so we're going to end every session with a student who's coming back from their as an undergraduate. And I think at least all of them are their minimum 3rd year in the program. But Sophia, I'll allow you to introduce Yaser.
It's my pleasure to introduce Yaser. We worked together this summer on comparing machine learning models and causal inference models with traditional causal discovery graphical models. He's going to talk about that. He has been fantastic. That's all I can say. It's been a hard program this year, and I wish we got to meet in person. But he has been fantastic, and he did great work. He was too fast for me, honestly, sometimes. And everything we discussed, he did like immediately. He's one of the most efficient people I have worked with, and I think you're going to enjoy his presentation.
Thank you. Let me share my screen now.
Perfect.
All right. So, good morning, everyone. I'm Yaser Morsi, and over the last 2 months, I've been working with Dr. Sophia Triantafilo at the Department of Biomedical Informatics at the University of Pittsburgh, and we've been doing comparative research with machine learning and causal models. So with the influx in observational data and the difficulties of performing controlled experiments in randomized controlled trials, researchers often analyze data with machine learning and can determine treatment impact and causal relationships depending on underlying statistical models with causal models. And in this study, we compare machine learning and causal discovery models under passive inference and compare causal inference and causal discovery models to see the strengths and weaknesses of each type of model on the same observational data sets. In doing so, we want to see if we're missing out on using causal graphs and see whether causal graphs can help us better analyze data and estimate treatment effects. So we're aware of the inherent strengths and weaknesses in regards to machine learning and causal models. One such difference is that some machine learning techniques don't make data distribution assumptions, and on the other hand, causal models that often make distribution assumptions. However, a strength of causal models is that they provide insight, causal insight to your data and interactions between variables and response variables. So we use 3 types of supervised machine learning models, discriminant analysis, support vector machines, and random forests. So with discriminant analysis, which you may know is a dimensionality reduction method, as well as a classification method, it increases separability across known categories by imposing an access depending on the number of categories. And support vector machines construct hyperplanes in high dimensional spaces and classify based on the best performing hyperplanes. And random forest is an ensemble method for classification that uses bagging or bootstrap and aggregation to make the decision, bootstrapping and using the aggregate to make decisions, which means that it selects random samples and predictors and creates multiple decision trees. Random forest and bagging help avoid overfitting data, which occurs when you could be using a small number of trees or a model made of a single decision tree. So finally, our causal models are based on causal Bayesian networks obtained through a fast greedy equivalent search. This algorithm takes in data and greedily searches over selected causal Bayesian networks and output the best model based on a Bayesian score. So it finds a model by improving the score by adding edges between nodes with forward stepping search and then removing edges with backward stepping, all to improve the score. So our resulting model is a directed acyclic graph that shows relations between covariates and a response variable, and in our case, a treatment variable as well. So to start, we have data provided by the Atlantic Causal Inference Conference. So this data contains 3,200 different data sets, 1,600 of which have a continuous response variable, and the other 1,600 have a binary response variable. And all of these were produced with 32 unique data generating processes. So each data set contains, like I said, a response variable and also a binary treatment variable. So whether a sample or an individual received the treatment or not, pre-treatment covariate. And each data set contains a different number of pre-treatment covariates. So another data set that we were analyzing was a data set used by Robert Lalonde from a nationally and privately funded work experience program, the National Supported Work Demonstration. So from this data, certain participants from socially and economically underprivileged backgrounds would get a training experience, which this work experience, whether they received it or not, would be used as the treatment indicator. Also in this data is a series of pre-intervention variables. some of which continuous and some of which binary. So age, the years of education, whether the individual was Black or not, whether they were Hispanic, whether they were married, if they have a degree, and the pre-intervention earning. And as the response variable, the data included their retrospective earnings for after the program. So with this data, we want to compare different inference approaches, passive inference and causal inference. So with passive inference, we're learning about the value of the variable when it's caused like in a normal way, which is just the causal Bayesian network that I mentioned earlier that we got with the fast gradient equivalent search algorithm. And under this, we're comparing machine learning and causal models, causal discovery models. And with causal inference, we're overriding the causal structure and we're intervening and controlling the treatment variable. The treatment variable. So this is represented by surgical intervention, where the arrow is directed into the variable being intervened on, which in our case is the treatment variable, are removed. We're comparing causal inference and causal discovery models. So like I said, under passive inference, we're seeing the performance of causal discovery and machine learning. So with machine learning, we utilized supervised learning algorithms mentioned earlier, LDA, QDA, support vector machines, and random forest to predict the response variable given the treatment and covariates on a test set. And with causal discovery, we do the same thing, but this time utilizing the causal Bayesian network obtained with the equivalent search for each data set. And these data sets are the 3,200 data sets as well as the nationally supported work demonstration data set. So from this causal Bayesian network, we extract the Markov blanket, which are the parents, children, and parents of the children variables with respect to the response variable. So we again predict the response variable on a test set after training the model. So we're essentially using it as a feature selection technique to get the more related features that represent the underlying mechanisms of the data. So under causal inference, we use Bayesian additive regression trees, or BART, to compare causal inference and causal discovery. So BART is a sum of tree models where each tree is constrained by regularization prior to avoid overfitting the data. And we control the treatment variable to estimate the average treatment effect. So BART is used to compare causal inference and causal discovery, and with causal discovery, we only use the parents of the response variable from the causal Bayesian network. And again, we're estimating the treatment effect of the treatment variable. So for the causal inference conference data that had a binary response, which were the 1,600 data sets, we used the area under an ROC curve to evaluate the performance of each model. So the larger the area under the curve, the better the model performs. So for each model, we plotted the AUC of each data set using machine learning, which used all features, or the causal discovery models, which only used the Markov blanket features. And we also included a line of equality for comparison. So if a point falls to the left of the line, then the machine learning model performs better. And if it falls to the right of the line, if more points fall to the right of the line, then the causal discovery models perform better. So as we can see, between all models, the performance of machine learning is generally better over the causal discovery models. And although the same is true with the support vector machine graph, it's much less pronounced than the other models, possibly because it may be overfitting the data or it might need tuning. So you also notice clusters of data sets and their performance in each models. So these clusters are between each on each model are the same data sets. So we see these same clusters that are similar in LDA, QDA, and random forest, but sometimes those same clusters, like I said, are more spread out in support vector machine, in the support vector machine model results. So we see a more inconsistent performance. So for the continuous response data, which were the other 1,600 data sets from the causal inference conference, we have a similar comparison, this time using the root mean squared error as the comparative metric. So the lower the RMSE, the better the model performs. So this time, the more values that fall to the right of the line of equality, the better the machine learning model was, and the more values that fall to the left, the better the causal discovery model was. And here we use linear regression and random forest for the continuous response variables. So we see a similar pattern to the binary response data where the machine learning model performs better as you see more clusters of data set to the right of the line. But when we look at random forest, we get better results overall, and most points fall closer to the line. So there are similar results between both models. This is possibly because the random forest is a more robust technique when dealing with features that may not be as informative as others. Because like I said, it selects the best subset of features for each decision tree and makes multiple decision trees to increase the performance. And finally, if we look at the NSW data under passive inference, we see a similar trend with the machine learning model outperforming causal discovery. And like the trend that we saw here with the ACIC data, we see random forest outperforming the linear regression model, whether it was with causal discovery or machine learning. So for each method, we also predicted the average treatment effect for each data set, which we can then compare to the true average treatment effect provided by the Atlantic Causal Inference Conference. And here we plotted a comparison of ATE predictions to the true ATE with a histogram. So we took the difference in the causal inference loss and the causal discovery loss. So the more values that are positive, the better causal discovery model is, and the more values that are negative, the better that the causal inference model is. So as you see, we have better performing causal discovery models when predicting the average treatment effect. So for the NSW data, we did something similar. We had the AT estimates using causal discovery and causal inference. And we also compared them to the proposed average treatment effect of the loaned. So with causal inference being slightly closer, there isn't much of a difference when we estimated the treatment effects. So distinguishing between the results of machine learning models and models based on causal Bayesian networks provided an insight on creating more robust models to better analyze observational data to gather treatment effects and predict response variables. So moving forward, we'd like to explore these implementations of machine learning and causal networks to yield better and more robust models. Thank you.
Excellent job, Yaser. I'm going to once again unmute everyone so we can give them a round of applause. All right, great work, Yaser. Are there any questions? We do have a question. All right, Yaser, and I muted you, so you're going to have to unmute yourself, Yaser. The question is, could you explain a little more about implementing machine learning to causal discovery?
So that was more of the exploratory research that we'd like to approach after this. What we focused on for the last 2 months was more of just the comparative research, just comparing machine learning and causal models. Yeah, like I said, in the future, that's what we want to explore and look at.
Excellent. Any other questions? And Yaser, I know, and again, correct me if I'm wrong, as I'm trying to do multiple things and maybe missing some things. You did this work, there were continuous variables, and then you also had it on discrete variables. Did you do it on any mixed data sets and compare causal discovery versus machine learning?
So with the response variable, we had either data sets that were either had a continuous response variable or a binary response variable. But for other predictor variables, yeah, they were mixed data sets, but for the response variables, no.
Excellent. And can you envision, I know you show machine learning in a lot of ways outperformed the causal discovery in these data sets. Are there data sets or data types do you think that the opposite might be true?
So we looked at, so what we have as the results are the general results for all the data sets, in total, but we can look at specific data sets from the ACSE, the ones that they generated, and look at specific ones and see that causal discovery could have outperformed machine learning, and we could explore why that happens. But for this presentation, I just included the general outperformance of machine learning to causal discovery, but there were a few data sets that the opposite was true, and we could explore why that was.
Excellent. Any other last questions for Yaser? If not, I want to again congratulate all of our first speakers. You all did an amazing job on a variety of different topics, which is always so fun. It's always so fun for me to learn about a lot of my colleagues' work across campus that I otherwise wouldn't get to see. So this is awesome. You guys all did a really wonderful job. And now we're going to take a break and we'll come back for session 2 at 10:30. This session will remain open the whole time. Feel free to turn off video, unmute, or sign out, sign back in, do whatever you want. But we'll be back at 10:30 for our second session. So thanks, everyone. Okay. So welcome back, everyone. Give everyone another 30 seconds to come back. All right, so welcome back, everyone. Again, getting ready for our second session. I want to thank all of the students who are about to present again and their mentors. In case the mentors weren't here, you're going to hear me thank them so much today. Again, giving of your time and dedicating yourself to students who you have never even met is remarkable given the current situation that we're living in. So thank you so much to the mentors. Thanks so much for the students. The first session went really, really well. We're going to start our second session now. And unfortunately, one of the students had an issue today and can no longer present, so Trayvon. So we will just have 4 presentations. So Erica, you will present immediately following Marcus. We'll follow the same format. For those of you who weren't here for the first session, we will hope that the mentor will introduce the student. The student will then give their 8 to 12 minute talk, followed by a few minutes of questions. All of the mentors and students should have privileges to share screens, share mics. If not, please let us know. But you can also use the participant button and raise your hand at any time if you'd like to speak, or use the chat feature in order to ask questions or give the presentation. For the students, one thing that I didn't mention, while you are talking, you may hear beeping. This happens when people are coming in or going out of the room. Ignore that. Solomon and I will take care of admitting people from the waiting room, so please don't let that be a distraction to you. So without further ado, I'm going to turn it over to Kieran. And is Jishnu here to introduce you, or someone to introduce you today?
It does not appear. I don't think he's come yet. Do you want me to just, I guess, like introduce myself?
You can introduce yourself. I mean, I can introduce you very briefly.
I would appreciate that. Yes, thank you.
Yeah, so Kieran, this is, and again, it's a little easier to introduce her because this is not her first time with us. So Kieran was with us in person last year, did an amazing job, was one of the brave students who agreed to be part of our first time piloting a year-wide program, which she did amazing at. So she lives in Georgia. And for those of you who don't know about our year-wide program, we try to take the work that the students do in the summer and then translate that into college scholarship opportunities or science fairs. And this is it's much easier for me in the Pittsburgh region because I know these. Outside of the Pittsburgh region, it really falls upon the students' shoulders. And Kieran went out, found competitions, did really, really well at the competitions, even though some of them became virtual, and had won some awards and did some amazing things. And we're so happy to have her back this summer in a virtual setting. And again, she's a student who initially, last year did wet lab work and came back to do some computational work too. So very well-rounded student, and very pleased to have Kieran here to talk. So Kieran, please.
Thank you so much for the introduction. So I'll begin. The title of my project is Univariate Analysis of Metabolomic Patient Data of Double Seropositive and Rheumatoid Factor Seropositive Rheumatoid Arthritis. So rheumatoid arthritis is a chronic inflammatory disease that affects around from 0.5 to 1% of adults worldwide. Additionally, it's also one of the more common autoimmune diseases, as you can see in this diagram, in relation to other autoimmune diseases, indicated by the length of the person, RA, which is also rheumatoid arthritis, which I will be abbreviating. And so whenever I say RA, that's the same thing as rheumatoid arthritis, and indicated by this long length of a person in relation to other autoimmune diseases. RA is one of the more common autoimmune diseases. RAs is classified under two broad categories. One is seropositive. Seropositive means that when you conduct a serology or a blood test, there are certain antibodies such as like rheumatoid factor that are found in those blood tests, in addition with other symptoms like joint inflammation. But in the second category, seronegative, these antibodies are not found in the blood, so it's mainly based on these symptoms like joint inflammation and from like imaging tests. So I will describe a little bit about how RA is diagnosed. So there's mainly 4 different categories. The first one is joint involvement, where physicians ask about symptoms like joint inflammation and use imaging tests like MRI and ultrasound to look for any bone deterioration, as well as then serology, which is important in that those seropositive RA that I discussed earlier, so they can look for RF, which is rheumatoid factor, which is an antibody found in the blood, and then ACPA, which is anti-citrulated peptide antibody, which also is indicative of seropositive RA, as well as some acute phase reactants and just the duration and how long these symptoms have been occurring. And a score of about 6 or greater is needed in order to diagnose with RA. So for seropositive RA, majority of the points come from the serology, while for seronegative RA, most of the points come from mostly joint inflammation. And so for seropositive RA, for a patient with seropositive RA, the patient can either have just rheumatoid factor antibodies, they can have rheumatoid factor antibodies and anti-CCP antibodies. They can have a numerous combination of these different antibodies, and they're still characterized under this broad category of seropositive. So one of the purposes of my project is to understand the differences between just, for example, rheumatoid factor positive patients and patients with rheumatoid factor and CCP positive patients. So, for example, when we're looking under the umbrella of seropositive patients, double seropositive would be both rheumatoid factor and anti-cyclic citrulated peptide antibody positive, which means they both have, for example, this is purple for rheumatoid factor and yellow. They will have both of these antibodies in their blood, while rheumatoid factor seropositive only has these rheumatoid factors present in their blood. So we're trying to understand the molecular differences. So we hypothesize that there are some molecular differences between this DFP, which I'm going to abbreviate for double seropositive, and then RFP patients, which I'll abbreviate for rheumatoid factor patients. And if we can, using these metabolites, we can develop predictive biomarkers for of these metabolites. So for example, when you go to a doctor or a physician, they use metabolites for other diseases and drug tests in order to diagnose for those. So if we're able to develop these predictive biomarkers and metabolites to distinguish between DFP and RFP, this could potentially create a more specified diagnosing procedure. And we will use an unbiased screening of 1,000 metabolites. So first, I used for approximately 45 RA de-identified patients, and these were treated with these 2 treatments, and which are used for RA. And then there was 2 primary data sets which I used. The first one was metabolomic data, so approximately 1,000 metabolites from liquid chromatography mass spectrometry. And then the second data set was serology data, which is important for distinguishing the different types of seropositive patients. So the first time point which I looked at was the baseline time point, which is before any of these 2 treatments. And looking at RF positive and CCP positive, I was able to classify those as DFP. And then with the serology data, looking at RF positive and CCP negative, I was able to classify that as RFP. And there was approximately 15 patients of DFP and approximately 30 patients of RFP. So with these baseline metabolites, I classified these baseline metabolites for these DFP and RFP. And I conducted independent and Mann-Whitney tests for each of these metabolites between the baseline DFP metabolite and the baseline RFP metabolite. Then at the second time point, which is the post-dose, which is after the treatment, I looked at the metabolites for DFP of the post-dose levels and then RFP metabolites of the post-dose levels, and conducted independent and Mann-Whitney tests for each metabolite between these two categories. So with the p-values, I wanted to see how many, approximately how many significant metabolites that I would be able to compare with. So with the t-test, these are the t-tests at the baseline, and these are at the post-dose, and the first row is the t-test, and the second row is the Mann-Whitney test. And the x-axis is the metabolites, which I number from 1 to approximately 925. And then the y-axis is the negative log of the p-values. And then this line, the blue line, is the threshold, which the threshold which I was using was 0.01, and negative log of 0.01 is 2. So this above this line would be the metabolites that are significant. So as you can see in all of these graphs, there are very few significant metabolites between the DFP and RFP for both t-tests and the Mann-Whitney test. So this is the t-test at the baseline time point between DFP and RFP. This I chose 2 of the most significant metabolites, and the x-axis is the mean RFP and mean DFP, and then the y-axis is the levels in the units are M over Z. And as you can see that it is significant by the error bars, but the problem is that I was doing multiple testing. So for each metabolite, I was conducting a t-test. So there's a correction which needs to be applied to when you conduct multiple tests. But unfortunately, when I corrected it, the Bonferroni and the Benjamini-Hochberg, I found that the p-values were no longer significant. And the same thing occurred when I looked at this at the post-dose visit 1 time point at the t-test with the same an x and y axis. And these were 2 of uridine and 3-urideopropanate were 2 of the most significant before the correction. But when you apply the correction, these were no longer significant. Then looking at the Mann-Whitney at the baseline time point between DFP and RFP, and I found also like sort of the same result. The p-values, this one had a significant p-value before the correction, but once I applied the Bonferroni and Benjamini-Hochberg corrections, they were no longer significant. And then with the Mann-Whitney test at the post-dose time point between DFP and RFP, I also found that the p-values were significant prior to adding the correction, but when I added these corrections, they were no longer significant. And like I discussed, the reason for adding these corrections was because of the multiple testing problem. So what we can conclude is that since they were no longer significant after applying the correction, instead of adding a univariate approach, we should add this motivates the generation for using a multivariate model. And actually, currently, I'm working with the undergrad Amna from TechBio, and we're developing a multivariable machine learning model to be able to better characterize the differences between DFP and RFP. And we're currently started working on random forest machine learning model, and we hope to receive some results from that. So some of the acknowledgements, I want to acknowledge my mentor, Dr. Das, for helping me throughout this project, as well as the postdoctoral associate, Dr. Rahman. I'd also like to thank Dr. Boone and Solomon so much for for doing this virtual program, especially, and giving me this amazing research experience, just like from last year, as well as Amna, who I'm working with, and then all the members of the Hillman Academy. Thank you. Any questions?
Excellent, great job, Kieran. I'm going to, again, unmute everyone so we can give her a proper round of applause. Good work. And are there any questions? And feel free to unmute if you're able, raise your hand in participants or ask a question in chat.
What makes those like correction tests, like, I guess, what makes them so stringent that none of the metabolites appear significant?
Okay, so what happens when you have a t-test or a Mann-Whitney test? When you're doing just one test, your p-value, let's just take 0.05. That 0.05 technically is, you're accepting, like, I guess, like a 5% chance that you could reject the null hypothesis, and that's called a type 1 error. So when you do so many tests, so I was doing 925 tests, the problem is that the type 1 error increases rapidly because you're doing the same test on the same set of data. So that's why you need to add that correction, because adding that correction accounts for that increased type 1 error. Otherwise, there would be a lot of false positives that happen. That's why adding that. That's why it's much better for me to add a multivariate sort of models to better be able to make conclusions from the data.
Thank you.
Excellent answer. Any other questions? Kieran, I'm curious if you or anyone else has ever done any quick visualizations to see if the features that you had, the 1,000 metabolites, grouped the patients into their two, I forget, the double positive and the other, either using all of the metabolites or using the most variable metabolites, doing a quick hierarchical clustering heat map or a quick PCA plot, if you'd see some good division between the two groups. Have you tried that, or have others ever tried to do that?
That's a great question. That's something I will look into and talk to my mentor, and hopefully, I don't know. I don't know much about that, but I can definitely look into that. That's a great question.
Cool. All right. Any other last quick question? All right. Really well done. Thanks, Kieran. So if there are additional questions, please continue to reach out in chat, and Kieran can answer you there. And for our next speaker, I will have, I think, Will, you're introducing Marcus, correct? Perfect.
That's correct. All right, so it's my pleasure to introduce his presentation. As we all know, this summer, the program has been a little bit different, and there's some unique challenges right now, but Marcus has really impressed me with his aptitude and his adaptability. He came into the program because of his interest in biology, but he didn't have much of an informatics or programming background at all. So I was very impressed when we had to go over some of the basics and the necessary skills for running our analysis, and he picked everything up so quickly from scratch, learning a lot of programming techniques as well as a lot of bioinformatics web tools as well. And on top of that, he was able to communicate and give us all of his data very easily by using different tools like Slack and Box and Zoom. It's hard when we're not in person, but it almost felt like we were together every single day through all this. So with all of that in mind, I think we were able to come up with a really cool project, and Marcus was able to answer a lot of cancer biology questions through all of these new things that he learned. So take it away, Marcus.
Thank you. So my project is the characterization of the tumor microenvironment. And the objective of this project was to see if there are any differences within the microenvironment across different cancer factors by identifying geo data sets, which I'll talk about later, that have cancer samples with these different factors I want to observe. And some background information you need to know is that understanding the tumor microenvironment will contribute to the overall understanding of tumor biology. and can help answer some questions such as why do some primary tumors metastasize and why do some patients respond to therapy. And as an example, response to therapy is a complex process that includes many of the tumor's properties, such as tumor burden and possibly the microenvironment. And the tumor using the Cancer Genome Atlas data, which I'll also talk about later. But this TCGA data doesn't always have good outcomes, and it has few metastatic samples. And so the tumor microenvironment is the sum of all the non-cancerous cells within and around the tumor. And at the right here is a model. And so there consists of stromal cells, which are the connective tissues within organs. And you can see some here, such as fibroblasts, and then immune cells, such as lymphocytes and cytokines. And how the tumor interacts with its environment is critical in its growth and metastasis. And so what I used was the Cancer Genome Atlas data, which was a project by the NCI, which sequenced like about 10,000 tumors and using a bunch of different genomic platforms. And at the bottom here, this is just a screenshot of how you download this data. And this is specifically the RNA sequencing data highlighted in red. And so RNA sequencing works by taking the total RNA within the cell, sequencing it, and it gets an output of bulk expression data. And then I use this with conjunction of gene expression omnibus datasets, which is a large database of published papers that includes data using TCGA. And so within this dataset, I chose 3 cancers: Kaposi sarcoma, non-small cell lung cancer, and gastric cancer. And each of these were looking at a specific factor. Kaposi sarcoma was looking at HIV-1's co-infection role in the tumor microenvironment. And there were 36 epidemic patients, meaning they had this co-infection, and 12 endemic, so they didn't. And small cell lung cancer, they were looking at a type of immunotherapy called anti-PD-1. And there were 5 responders to the therapy and 11 non-responders. In gastric, they were looking at the tumor microenvironment differences of primary and metastatic cancers, and even 50-50 split of these samples. And to the right here, you can see a screenshot of how you download this data. And both TCGA and GEO are publicly available data sets you can easily download. And so I then use bioinformatics tools that use both marker gene approach and deconvolution. So marker gene qualifies cell types based on a gene set derived from target transcriptomic studies, and deconvolution calculates cell types per sample from regression on a matrix of expression profiles. And so one of the tools I used was CyberSort. It's a web tool that uses deconvolution. Excel, which is another web tool that uses marker gene approach. And then lastly, Estimate, which was an R package that uses the marker gene approach. And on the right here, you can see the workflow of Estimate. So it starts with the TCGA data and also the public database data, which I got from GEO. Starts with the common genes, goes all the way down until you get a stromal score, estimate score, and immune score. So the stromal score is how much stromal cells there are. So the higher the number, the higher amount of cells. Immune score, which is the presence of immune cells, and estimate score is the tumor purity. So the higher the number, the less cancer cells there are present overall in terms of relation to stromal and immune cells. And so for my results, you can see in these 2 graphs here that the primary seems to have a slightly higher stromal cell score in estimate, but Excel, it seems to be about the same. But you can see in these 2 graphs here that the metastatic have a higher immune score, but overall they have about the same microenvironment score, suggesting that they have similar tumor purities. And then my results for lung cancer, within all of my graphs, you can see in the right columns here that the responders to the anti-PD-1 treatment had higher stromal, immune, and overall microenvironment scores. And this was kind of expected, just because of my hypothesis that an increased number of immune cells will create a higher response to immunotherapy. And then lastly, I looked at Kaposi sarcoma. And in these two graphs here, it shows the immune score. And so endemic patients, which were the ones without the HIV-1 co-infection, had higher immune scores, which is also to be expected because HIV-1 does have an impact on the immune system. And endemic overall had a higher microenvironment score, but it had relatively about the same stromal score with estimate, but a slightly higher one based on Excel. And you can conclude from this that different clinical groups seem to have different tumor microenvironments. And while these results are promising and intriguing, you have to take into account that these were small sample sizes and different tools don't always produce the same results. And there were plenty of other variables that could have had an impact, such as tissue processing. So in future studies, there needs to be more carefully controlled, especially with tissue selection and just larger study sizes in general, using and then using this data from these larger studies, and then also clinical trials, so you're able to subset these data based on different factors within the cancer. And I specifically want to subset more anti-PD-1 treatments because that's what I'm most interested in. And I just want to take the time to acknowledge Will and all the time he took to help me throughout the summer. I came in knowing nothing about the topic, and he took a lot of time to teach me, as well as Uma. She helped me a lot too. Dr. Boone and Solomon and everyone who made this summer possible. I really enjoyed it. That's it.
Excellent. Great job, Marcus. So I'm again going to unmute everyone so we can give you a round of applause. All right, great work. Are there any questions for Marcus? Again, feel free to unmute if you can, raise your hand or ask in chat. So Marcus, I think I'll start it off. So you tested these two different algorithms. Do you have any sense, after doing this, and you did a lot of work, of which algorithm might be better than the other? Is there one that you would recommend to someone coming into this work again, one over the other, and for what reasons?
I don't think you can really pick which bioinformatics tool is the best, just because they all use different approaches, and there's no like one set algorithm that will get you 100% accurate results.
Yeah, you're probably right. As much as we would like that not to be the case, and there to be a really good tool at doing this because it's such important work. And do you know, and if you don't, that's totally fine. When these algorithms were published, did any of them do comparisons on bulk RNA-seq versus single-cell RNA-seq of similar or the same samples?
I'm not entirely sure. I know I use bulk RNA sequencing for them.
Right.
But...
David, this is Uma. If I may interject, I'm also not sure that they compared the algorithms directly, but I know that CyberSort has been, there's a CyberSort for single cell version now.
Yes, that's a lot of these tools are trying to be updated now that single cell is becoming more and more prevalent. They have to change everything because they have to take into account these UMIs, but a lot of the tools are coming out now with single cell versions.
Very cool. Any other questions for Marcus? So actually, Marcus, I have one other question, unrelated. So you came in with a strong interest in biology. I think originally when you applied to the Hillman Academy, were not interested in the computational sites as much. Do you have now a different perspective or appreciation for the kind of the power and what we can do? And maybe that was the biased way to ask that question. So what are your thoughts now on computational research, coming in with biology as your main interest?
I definitely have an appreciation for it, but I don't know. I don't know. I feel like what I'm more. It's just a lot.
Yeah, that's totally fair. All right. So great job, Marcus. And now we're going to move on to Erica. And I don't know, Casey maybe is introducing you, Erica?
Yeah.
Perfect.
Yep, that's me. First, I just want to thank Dr. Boone and Solomon for setting up this really great program and also for adapting so well to the kind of change in circumstances. So this summer, Erica has worked with me and Natasha Miskov-Zivanov in Natasha's Melody Lab using a tool that automatically compares existing biological models to relevant published literature. And this project is actually the compilation of about 6 or 7 years worth of work. And Erica was able to kind of get up to speed and be using it and understanding it in just a couple of weeks. So she's done an amazing job. She's learned a lot. I've learned a lot from her, from the questions she's asked and from the work that she's done. And I'm really excited for all of you to see her presentation. So turn it over to you, Erica.
Thank you. Okay. So hello, everyone. My name is Erica Rispoli. My mentors are Casey Hansen and Dr. Natasha Miskov-Zivanov. My project is titled A Corroborative Approach to Understanding Model Curation and Extension Using Violin. So machine readers are extremely useful in biological event extraction. So a machine reader can read through a paper, optimally a paper about a specific pathway or system, and then the machine reader can extract specific interactions, usually interactions between proteins. The machine reader that I used was called REACH. So in this figure is a representation of a single extracted interaction. You can see in the blue that you have the regulator, which is AKT. And then between them, you have the interaction, which is phosphorylation. So AKT phosphorylates MDM2, which is the regulated node. You can also see that the machine reader pulls attributes. So that's up here and down here. So since machine readers can decrease the time spent tediously extracting mass amounts of interactions from biomedical literature, there are tools that have been created to kind of supplement this process. So the one that I'm working with is Violin. So Violin is a tool that can intake a machine reading output and a model and then classify and compare the interactions. So with Violin, you can compare models and either extend or add details to these models. So this is a figure about how Violin fits in with the overall machine reading process. So you have your search queries, which is basically where you're specifying what you want your papers to be about. You curate that into a reading set, which you then put into the machine readers, which then extract the interactions, and you get your LEEs, which is just like your list of interactions. You put that along with the baseline model into Violin, and you get the classified and scored LEEs, which you can then use for model assembly. This figure is very similar, but it also represents the interaction class, the interactions and the scoring system in the classifier. So Violin assigns different score values, each representing different categories and their level of significance. So there's the epistemic value, which measures the confidence that a given interaction was actually pulled by the machine reader correctly. And then there's the match score, which measures new introductions from an LEE. So there are several cases here. So your first case is that you introduce a new regulated node, and that's example A. So you can see Y is the new node, and it's being regulated by a pre-existing node, and that would have a match value of 1. And then another possibility is that you could have the LEE introduce a new regulator, so that's example B. So you can see that the new node is regulating a pre-existing node, and that would have a match value of 100. And then C is an example of both a new regulator and a new regulation. So you can see that Y is the new regulated element, and then X is the new regulator element. And that would have a match value of 0.1. And then finally, you have example D, where there's neither a new regulator nor a new regulated, but there is a new connection between two pre-existing nodes. There's also the chi-score, which is a numeric value representing the different categories of comparisons and classifications that Violin can find. So you have corroborations, extensions, and contradictions. And all of these categories and values together can allow us to efficiently and accurately compare mass amounts of biological literature to existing models to either validate or extend the model. So the purpose of my study is to use Violin in a corroborative way where I compare an existing model of the BDNF to NF-kappa B pathway to its own reference literature. And the idea behind this is that the different scores and classifications can help me understand this original model and kind of determine what the authors wanted to emphasize in this model. And this can hopefully reveal the major connections that may contribute to major depressive disorder. So this is my chosen model. I got this model on the NDEX database. It's a model of the BDNF to NF-kappa B pathway, like I said before, specifically in the context of major depressive disorder. So this model contained 72 nodes, which represent the proteins. So those are these right here, and 82 edges, which represent the interactions between those proteins. So these are the interactions. So in mood disorders, if you look at changes in the brain, they're often marked by a decrease in neurogenesis, along with an actual alteration of synaptic structure and a decrease in neuroplasticity. And all of these are affected by levels of BDNF. So in mood disorders, you can notice a drastic decrease in BDNF, and thus these effects occur. Like BDNF, NF-kappa B is also linked to affecting neuroplasticity. So that's why NF-kappa B is focused on here. So overall, the mechanisms that regulate BDNF are very important to influencing MDD, since it's a biomarker for MDD. So that's why this model is mainly focused on those two proteins. So first, I chose a model based on accuracy and also necessity for extension, since I plan to further this project, which I'll talk about a bit later. And then I gathered the PMCIDs from all the reference literature of that model, which I then inputted into Indra, which basically assembled those papers for machine reading. So that's right here. And then I used Reach to extract the interactions from the 30 reference papers. And then I went over the reading output for formatting, and I checked for reading errors. So this is a couple rows from the reading output. You can see that the regulated element here is gene expression, which is a bioprocess. So it pulls from the gene ontology database and is given a gene ontology database ID. And you also have the regulator, which is BDNF in this case. And then you also have the evidence text, which is the sentence that the machine reader pulled this interaction from. So then I formatted the model into the biorecipes format for compatibility with Violin. And then I ran Violin on both the reading output and the formatted model, which gave me the comparisons between them. And then finally, I used an analysis script to further investigate the Violin output. And then I used Cytoscape to convert the reading output in the extended model spreadsheet into static models, which you can see here. So the one on the left here is just the reading output interactions. So notice that there's the main cluster and it kind of branches out. And then the model on the right is the graph of the original model with the Violin extensions added on. So you still have this main cluster, and the interactions that are connected to this cluster are going to be more significant and have a higher total score than the interactions that are kind of floating around out here, which are going to have a lower significance and a lower total score. So here at the bottom is a row from the Violin extensions file. So you can see that this interaction has an evidence score of 2, meaning that across all the literature that I put into the machine reader, this interaction was mentioned twice. It has a match level of 100, which means the LEE introduced a new regulator, which in this case is NAF oxidase. So that's an example of kind of how you'd use the scores to analyze a specific interaction. This top table shows that the match score was mainly around 0.1. So you can see that around 100 interactions had a match score of 0.1. And what this means, this indicates both a new regulator and a new regulated. So that makes sense why we're getting a lot of those as extensions now, because those are kind of the floating interactions that you saw earlier. So they're not very significant. Therefore, they were left out by the creators of the model. And that's why I'm getting them as extensions now. And then the bottom graph is the total score. So the total score contains all of the previous scores. So the match level, the match score, the kind score, and then the epistemic value. So you can see that you mostly have total scores between 40 and 50. And this is because since we have a majority of match scores that are 0.1, and since match scores of 0.1 are extensions, they're given a kind score of 40. So you get interactions mostly in the 40.1 to 50 range. So the comparisons between the original model and the reference model provide insight into the reference influence on the final model, which can reveal these major connections that may contribute to MDD. So analysis revealed that Violin found 163 extensions, meaning that there were many interactions from the reference literature that were left out, but the interactions that were left in mainly involved BDNF, NF-kappa B, and TRK-B, which displays that these 3 proteins were deemed significant by the authors of the original model. Violin found 5 corroborations, all but 1, in interactions containing BDNF, which just further emphasizes BDNF's importance to the system. And then finally, Violin found 1 contradiction and 1 phosphorylation contradiction. So the first contradiction can be attributed to a reading or formatting error, and the second contradiction happened because machine readers assume that the phosphorylation mechanism is positive, when in this case it was negative. So the reading output was saying that the interaction was positive, and the model was saying that it was negative. So Violin was able to provide further insight into the index model of the BDNF to NF-kappa B pathway. Specifically, the Violin comparisons denote which interactions were deemed significant to the original model. So Violin can enhance our ability to quickly extend, validate, and develop models of biological systems, which can allow further understanding of complex systems in their mechanisms. Violin's scoring system can help us understand which interactions are most significant to the system, and automating this whole process will allow for efficient comparison and expansion of many types of models. So this project was kind of a first step, and I wasn't able to do as much as I was planning to do. So for further research, I plan to create my own reading set that's specifically tailored to extend a specific area of the model that I worked with. So instead of just comparing it to its own reference literature, I would be comparing it to a reading set that I've curated. So thank you so much to Casey Hansen for all her mentorship and guidance throughout this project. She was an incredible mentor, and I'm so grateful that I got to work with her for these past 7 weeks. Thank you to Dr. Natasha Miskov-Zivanov for all of her feedback and support. She really kept me on track, and she had great feedback. And then thank you to the whole Hillman Academy. The students that I got to work with were really amazing, and it's been great to hear all their talks today. And especially thank you to Dr. David Bone and Solomon Lifshitz. I know how easy it would have been to just say, Hillman's not happening this year. So I'm really grateful for to be able to do this program and all of the opportunities that it gave me. So thank you. Are there any questions?
Excellent. Great job again. You know the drill. I'm going to unmute everyone. Let's give her a round of applause for such a great job. Excellent job. Any questions for Erica?
So this is Malavika. I have a question. So did this model pick up any molecules, like in the same pathway? Kind of, you mentioned about NF-kappa B. So, like this NF-kappa B pathway has many other proteins in the same pathway. So did it also pick up any other at the same time? Or did you see anything?
Erica, you need to unmute. Sorry.
I couldn't find my little icon. I think the model that we worked with was only proteins and like biological processes. I don't think there were any molecules that were a part of the either the reading output or the original model that I got off Index. I hope.
Okay, thank you.
Any other questions? And Erica, I again, feel free if you don't know this question, and I may have asked you this before, as I'm always trying to learn more about these machine readers and the literature that goes into them and what comes out. Do you know what evidence does the machine reader look for to say that there is a connection? Is it based on very specific text? Is it based on evidence from specific assays? Is it just them being mentioned in the same report together? Can you tell me a little bit about how, and again, I know you're working more with Violin and creating the cytoscape afterwards. If you don't know, I completely understand. But I'm just curious of this, the results you're putting into Violin to create your model. What exactly does that look like? How is that curated?
Okay. Yeah, I'm not an expert on machine readers by any means, but I'll try my best. So I think that the machine reader is looking for mainly like regulates or is regulated by or phosphorylation and then those the proteins that are mentioned with that, I think.
Great. Okay. So it's,
Yeah,
it's
I could.
Go ahead, Casey. Sorry.
Yeah, I could, just because that was kind of, we zipped over that just because machine readers themselves are their own project. But that's basically it, is they have, I call it, it's like they do some kind of sentence parsing. And they have specific words that they look for that they say denote specific types of interactions, like translocation or post-translation modifications, or directed relationships or bindings. And they basically have a dictionary of these words mean an interaction happens.
Cool. Great. Any other last question for Erica? If not, of course, you can continue to ask her questions through the chat. But we will move on to our last talk of this session, who's from another one of our third year returning, I think third year, maybe fourth year. I can't even keep track anymore. I think it's just third returning students. And I think I saw Chakra come in. Chakra, do you want to introduce Yash? If not, I'm happy to do so as well.
David, go ahead.
Okay, so again,
because I know Yash had a much longer connection with the academy, and yeah, and it was great to have Yash in this summer, to say the least. Yeah,
great. Yeah, Yash has been with us for several years now. Is originally from New Jersey, now at Vassar. He's again, it's like Yaser and Neil, who will come later, and Taylor. It's so fun to see students progress through years of the program, of like where they started and where they are now. Yash is no exception. He's been fantastic to have in the program, has been a critical part of the program the last several years, has done now a variety of different types of research too, which is something that I always try to encourage. And he's been great. There's a family history. His brother was in the program too. So we've known the Patels for now quite a long time, since I basically started this in 2015 of taking on this role. There has been a Patel in the program ever since. And I hope that continues. I hope that continues for a lot of the younger siblings that I've seen on camera over the course of this summer. I hope that you guys are all feeding them into the program. Yash, it has been amazing, so thank you. I know the only downside is I didn't get to beat Yash playing basketball tons of times this year, like the last couple of years, but that's okay. Hopefully we'll meet again in person. So Yash, please take it away.
Thank you guys so much for the introductions. So my project this summer revolves around finding recurrence-associated phenotypes in colorectal cancer data classified by an unsupervised learning algorithm for characterizing cellular phenotypic heterogeneity. So colorectal cancer is the third most common type of cancer in the world, and it can be divided into 4 main stages, with the 4th stage having a survival rate of about less than 5%. Recently, advances in multiplex imaging technology has allowed us to better be able to analyze tissue and tumor samples. And so instead of only being able to use one biomarker per tissue sample, we are now able to use many biomarkers on the same tissue sample. And this process allows us to better analyze the spatial relationships in the tumor microenvironment. An example of one of these technologies is CellDive, and it's able to use almost over 60 biomarkers for one tissue sample. So the tumor microenvironment of colon cancer is very important in determining disease progression and prognosis for patients. And currently, algorithms and software that are used to analyze hyperplex images are not supervised, and they lack the ability to identify rare cell populations. However, my lab created an algorithm called LEAF, which is an unsupervised learning algorithm for characterizing cellular phenotypic heterogeneity. And what it does is it takes in hyperplex images of tumor samples as input, and it's able to construct computational phenotypic trees like you see to the one to the right. And unlike other software, it's able to recognize rare cell populations, transition cells, and in general, cells that are linked with CRC progression in cancer biology, so we can better treat patients. So my data set consists of 213 CRC tissue samples that are all hyperplexed using cell dive technology with exactly 51 biomarkers. And within the data set, we also have various clinical variables. So we use various statistical analyses to predict 5-year CRC recurrence. The first was multiple logistic regression, and then we used Kaplan-Meier curves to further analyze the significance. And finally, we used some minor correlation analyses. So for each level of the hierarchy that I showed you guys before, we ran a multiple logistic regression model to predict 5-year recurrence of CRC. And as the explanatory variables, we used the sum of the ownership probabilities for each phenotype of the patients. And for the last 2 levels of the hierarchy, when we ran the model, we found that both phenotype 7 and 8 were consistently significant. And these were associated with signaling pathways and oncogenes. And so as you can see in the table, highlighted in red are the p-values for phenotype 7 and 8, and they are indeed significant. So then we wanted to further explore phenotype 7 and 8, and we wanted to see if the extremes of the phenotypes were significant. So we used Kaplan-Meier curves, and we grouped each phenotype into 3 groups. So the first was group A, which was the bottom 20% of the phenotype, group B, which is the middle 60%, and finally group C, which is the top 20%. As you can see, we used a log rank test to see if the distinction in the curves were significant. However, neither of these 2 phenotypes were significant in terms of their extremes, but phenotype 7 was phenotype 7 did have more distinct curves than phenotype 8 in favor of group B, which was the middle 60%. So in conclusion, using all 51 biomarkers, LEAF was not able to significantly predict CRC recurrence in 5 years. And in the future, we'd like to increase the sample size of our data. 213 is a pretty small sample. We'd like to expand the usage of LEAF to other cancer types and see what it can do. We'd also like to explore combinations of different biomarkers for analyses. So for example, instead of using all 51 biomarkers, we could use a small subset of biomarkers that are targeted at immune cells, and we could see if those have a statistical significance when it comes to predicting 5-year CRC. And we'd also like to improve the LEAF algorithm for better classification, and finally include spatial statistics for CRC classification. I'd like to thank my PI, Dr. Chakra Chanabola. I was very lucky to work with him this summer, and my mentor, Samantha Furman. I'd also like to thank Dr. David Boone and Solomon Lipschitz for making this summer possible for me. And finally, the Doris Duke Charitable Foundation. Thank you.
Excellent job, Yash. Going to unmute everyone, and let's give him a round of applause. Excellent job. Any questions for Yash? Again, feel free to unmute, raise your hand, or ask in the chat. So Yash, I'll ask a quick question. So in your samples, how many patients did you run this on? I can't remember. You can unmute. I have you muted, sorry. There you go.
113 patients.
113. So when you did your breakdown for the Kaplan-Meier curves, you did the top what percent and bottom, like top 10 and bottom 10%?
Yeah, here's a summary of the data set. I did the top 20%, the bottom 20%, and the middle 60%.
Okay, and this is actually where I was going with this question. I was curious if you noticed between those data sets where there are already clinical variables like stage that might drive the division of the patients into those rather than the features you're selected? And if so, if you have a large data set, can you maybe do it all in stage 2 tumors to see if you can do some predictions that way?
Yeah, we could. We actually did do some Kaplan-Meier curves subsetting within the stages, but we wanted to. My summer revolved around running it on the entire data set.
Cool. Yeah, because that could always, like you said, you were interested in combining covariates and
I wanted
combining
to see
things. Yeah.
how I could group the patients to see if they could predict 5-year recurrence.
Very cool. I think it'll be cool in the future whenever this is done, also taking into consideration stage and other clinical features that are known of CRC and others too.
Yeah, of course.
Oh, we have a question. Solomon, can you unmute Sunny? I can't do it.
Hi, yes,
Oh, perfect.
I can. Thank you very much. Great talk. I'm very interested in that. So on last slide, you mentioned the spatial statistics. Can you elaborate a little bit more about that? What does that mean?
Yeah, so for my study, we didn't use spatial statistics. We just use the ownership probabilities for each patient. But we'd like to use, for example, we could say if one specific phenotype is closer to another one, then it might be it might better be able to predict 5-year recurrence. One is basically spatial statistics is whether or not a certain phenotype is close to other phenotypes and in their proximity.
Yes, absolutely. That would be amazing. Thank you.
You're welcome.
Excellent. Any other questions? If not, again, I want to congratulate the students. A really, really amazing job. Second session, just like the first session, you guys did amazing, amazing work, amazing presentations. Thanks again to the mentors who spent so much time this summer working remotely with these students, and obviously to great successes. So congratulations, session 2. We will now take our lunch break. There is, we have catered food outside. Just kidding. We normally would. I wish we could have done it for you again this year, but you can go now. Please try to get lunch and come back for our third session, which will start at 12:15. So thanks again. Again, this channel will remain open for those of you who weren't here before. It will remain open all day. So you can leave, come back in, or just turn off your video and mute. And we will start back up at 12:15. So thanks everyone, and congratulations again.
All right, thank you everyone for coming back. We'll just be starting in a minute or two.
Welcome back, everyone. And welcome to those of you who are joining us for the first time. Again, for the students and for the participants who've been here for both the morning sessions, I was absolutely amazed at the quality of the work. So congratulations again to the students. Thank you for your persistence and for being willing to work during these difficult times. And again, so I'm saying this over and over again because I thank both the students and the mentors. It would have been so easy to tell me that you weren't able to take a student this year, given that labs were shutting down and everything that was happening. And I really, really appreciate the work that you've done. And again, I'm absolutely amazed at the quality of work that these students have been able to do in less than 7 weeks. Some of them coming in with computational interest, some without, and your work with them has been phenomenal. So I really appreciate you and all that you've done. And so again, just to explain logistics for those who are just coming in, I will turn the mic over to the mentors in order to introduce the students. The students will talk for 8 to 12 minutes, followed by questions. You feel free to ask questions either by unmuting. If you can't unmute, ask Solomon, who is the host, to unmute you, or raise your hand in the participant box, or just ask the question through chat. And then we will move forward. We have 5 great talks lined up for this session, and let's get underway. So Amanda will go first, and I don't know, Steffi, are you introducing her, or Vasiri?
Yes, I'm introducing her. I have the honor of introducing her. So very briefly, so again, my name is Steffi Österreich. I'm a professor here in the Women's Cancer Research Center working on breast cancer. We have had the tremendous pleasure to host Amanda Faber this summer throughout the academy. So briefly, Amanda is academically doing phenomenal, like I think most of the scholars do. I think Amanda has many, many awards, and I really don't want to go through all of them. Maybe just highlight a few in that she has had perfect score on the FSA. She is she got numerous awards for her activities in singing. So in addition to being academically very interested and very driven, she actually is very interested in singing. She has been in the choir more than 8 years. She is doing sight reading, and has won numerous awards for this. And I was super delighted because I was almost joking, saying, Amanda, maybe you can sing something in German. And then the next moment she sends me a link to a YouTube video where she recorded a German choral singing, which was like absolutely phenomenal. So I thought it was really, really cool. So in addition to her academics, her choir, her singing, she's also engaged with the community. So she is helping Latin American kids. So I should actually say Amanda's in Florida. She is in West Palm Beach in attending the Dreyfus School of Arts there. So she's member of the Spanish Honor Society, works with Latin American kids. And so pretty busy schedule. So now about the summer, I should say that, like, I think many mentors, and they've just said that, I think when we originally debated how does this work, we were a little bit nervous about this. And then Amanda joined us, and I think it couldn't have gone much better. I think it worked really, really well. Amanda basically did 2 things. She learned R throughout the summer, and she worked on a project which she will share with you, where she made progress and was absolutely instrumental in helping us to drive the project forward. We hope she will actually continue to work with us over the next months and year to work on this project. And like you will hopefully see in a minute, it has been really very, very productive. And, against our original concern, this was absolutely fantastic. I would like to thank Dr. Vasiri Lee, who is on the phone, who's not on the phone, but I mean on the computer, I think, who was her main mentor throughout the summer. But in addition to Vasiri, I would also like to thank Dorothy, Carter, and Yang Wu, who also mentored her. And then obviously Dr. Jennifer Xavier, who oversaw a lot of it. So we had a great summer. We were very, very delighted to host Amanda. And I would like to thank all the other guys in our group who mentored her, and then obviously Dave and Salomon for running the whole absolutely outstanding program. So thanks everybody, and Amanda, I give it over to you.
Thank you so much for that really kind introduction. Okay, so good afternoon, everyone. My project is titled Curation, Metadata Analysis, and Integrative Concordance Analysis of NGS Datasets for Implementation in a 17-beta estradiol or E2 treated database. So just for some introduction and background, I'm going to tell you about the binding of estrogen to the estrogen receptor and how it regulates cell proliferation. So 17-beta estradiol or E2 is a common type of estrogen or hormonal steroid. It has many physiological roles, such as regulating the menstrual cycle, regulating bone density, and triggering the proliferation of cells in mammary tissue. And E2 is what's called the ligand for the ER, or the estrogen receptor. So it basically binds to the estrogen receptor. But too much expression of the estrogen receptor is harmful, because as you can see in the picture here, like when the estrogen binds to the estrogen receptor, it triggers cell proliferation. But when there's too much of that estrogen receptor, there's in turn too much cell proliferation, which leads to tumor growth. So here's a figure that shows the effects of an important and widespread anti-ER drug. It's called tamoxifen. So basically, when you add high dosage of tamoxifen to cells that present the estrogen receptor, it competes with E2 to bind to the ER. And ultimately, it will win and block the binding of E2 to the ER, thus inhibiting proliferation of mammary tissue cells. So next, in order to analyze and just display the expression of the estrogen receptor in breast cancer cells, we need what's called next generation sequencing. So RNA-seq is a commonly used NGS technique. It allows us to infer the proteins that certain binds, for which certain genes code for. So it shows researchers genes that are differentially expressed among cells that receive different treatments. So let's say you have an untreated cell and a cell that's treated with E2. You'd isolate its RNA. RNA is just an intermediate between DNA and proteins. So RNA tells us what genes in the DNA will actually be coded into proteins. And you sequence the RNA, and you're able to use those sequencing reads to do some analysis of what genes are being expressed. And for example, I just inserted a volcano plot that I made that shows how you can visualize differential expression. So another NGS technique is called ChIP-seq, or chromatin immunoprecipitation. It indicates the genes to which a certain protein binds. So pretend that these blue ovals are the estrogen receptor. In order to immunoprecipitate the DNA, you would add an antibody, an ER antibody. And these peaks show on which regions of the genome does this ER antibody like agglutinate most often, and that will in turn tell you which genes a protein binds to most. So I split my project into 3 categories. The first category was curating various data sets for implementation in an E2 treated breast cancer cell database. So the E2 treated database team found approximately 230 E2 focused data sets that use these NGS techniques. And my job was to curate them, verify information was correct, and add some information to supplement what they found and put in an inventory. So the next step was analyzing metadata of some of these data sets, most notably RNA-seq and ChIP-seq experiments. And then thirdly, I performed direct integrative analysis of RNA-seq and ChIP-seq data. So that basically entails directly overlapping RNA-seq and ChIP-seq data that was generated in the Lee Osterreich lab. And the purpose of this was to examine genes that are shared among both sequencing methods. So the first step was curating data sets for an E2 treated breast cancer cell database. And I made this flowchart that shows the process. So you start at the gene expression omnibus, and you have certain key terms to find these E2 treated, E2 focused data sets like E2, estrogen, estradiol. And once you find data sets with one or more of those key terms, you determine whether it's an actual E2 treated, because some aren't, some are a little misleading. But if they are a data set that is pertinent to this kind of research, you enter a lot of information about those data sets in the inventory. For example, the number of biological replicates used, or the publication information, the geo-accession number, which allows you to find a specific data set and download the data directly from the gene expression omnibus. And these columns just give more experimental conditions like the treatment duration, the existence of a negative control, and the drug concentration, and like whether they used E2 or anti-ER drugs such as tamoxifen or fulvestrant. So the next step was metadata analysis. So the first kind, I generated many metadata analysis plots, but I'm only showing you my favorites out of the interest of time. So the first plot I generated was used in ChIP-seq experiments and those in RNA-seq experiments. So there's only one replicate used most commonly in ChIP-seq experiments, whereas in RNA-seq and microarray experiments, they're between 2 and 3. Additionally, I analyzed the E2 dosages. So most commonly, there was an E2 dosage of 10 nanomolar in ChIP-seq and in RNA-seq. And similarly, this is a factor that I analyzed that was shared among both experiment types. The most common cell lines used in both experiments was either MCF7 or T47D. But the treatment durations used in both kinds of experiments had extensive variability. So in order to do the last step of my project, which was integrative analysis, I first had to learn how to use R to do this genomic analysis with RNA-seq and ChIP-seq data. So these are two examples of plots that I generated based on an RNA-seq data set. A PCA plot basically allows you to cluster different samples based on gene expression. So here you can see the E2 cells, E2 treated cells were clustered, and the control cells clustered here. And a volcano plot basically allows you to see differential gene expression and how statistically significant that differential gene expression actually is. So then I analyzed ChIP-seq data. I overlapped binding sites of the estrogen receptor in MCF7E2 treated cells and the control cells. And I did the same in another common cell line. And then with this information, I was able to overlap both cell lines that were treated with E2. And as you could see, like the overlap really isn't significant in comparison with the sheer amount of binding sites for the ER and MCF7 cells. So now it was time for the direct integrative analysis. So basically I overlapped upregulated differentially expressed genes in both of these cell lines with differentially binding genes. And you can see that the overlap ratio is higher in MCF7 cells than it is in T47D cells. But you see something a little different in the downregulated. In T47D cells, there's a much smaller overlap ratio. But in the MCF7 cells, the overlap ratio of downregulated differentially expressed genes and differentially binding genes was actually slightly greater than that of the upregulated overlap genes. So here's some takeaways you can draw from this data. MCF7 cells contain more differentially binding genes than do T47D cells, making the MCF7 overlap ratio much greater. And when you apply statistical tests, you find that the proportion of T47D upregulated genes that contains also the differentially binding genes from ChIP-seq this is significantly higher than that of the downregulated genes. However, the proportion of MCF7 downregulated genes that contain the differentially binding genes are not proven to be significantly higher than that of the upregulated MCF7 genes. So here is some discussion and future directions for the database. I'm going to be adding more data sets, maybe from a source other than GEO. And I will, the inventory will also be released to the public in conjunction with the web platform database that the E2 treated database team within the lab will be releasing. And basically just for the overall significance of the database, it will facilitate reproducibility of experiments requiring genomic data of this nature. And the second part of my project, metadata analysis will be expanded to describe data sets of every NGS method. And in the future, a Shiny app or a website from a local computer server will be created that allows users to customize visualization settings for various metadata. And lastly, here's some limitations of the direct integrative analysis I performed. I should definitely overlap several more E2 treated RNA-seq and ChIP-seq data sets. Different thresholds should be set, and more descriptive algorithms can be used. And lastly, I will do concordance analysis within individual NGS methods to see similarity of data. So I'd like to acknowledge Dr. Jennifer Xavier, Dr. Adrian Lee, and Dr. Stephanie Osterreich for giving me such consistent support and feedback, and you guys really made me feel special. And Dr. Zeki Lee, Dorothy Carter, and Yang Wu, you guys taught me the foundations of genomics and bioinformatics and coding in R, and you guys always answered all of my questions. And lastly, Dr. David Boone and Solomon Lifshitz, you guys made this virtual research experience possible for me, and I could not be more grateful. Thank you.
Excellent job, Amanda. So I'm going to unmute everyone so that we can give her a proper round of applause. Excellent work, Amanda. And now we have time for a couple of questions, if anyone has any. Again, feel free to unmute, raise your hand, or ask questions in the chat. And while we're waiting, Amanda, I have a question. So did you actually recall all of the ChIP-seq binding locations or the differentially expressed genes? Did you download raw data and run them through the platforms, or were you using, I guess, what was the input that you were using whenever you were curating this?
So for the RNA-seq data, I used the R package DE-seq2 to generate a differentially expressed gene list with certain thresholds that I set. And I did the same thing with ChIP-seeker, the R package ChIP-seeker, to basically create a gene list with the significant differentially binding genes.
Oh, that's cool. So you did redo a lot of the work. Did you see, and again, from study to study, there may have been variation between, and I don't know, maybe there wasn't, between the aligner that was used, between the read lengths of the RNA-seq. Did you do any kind of regression analysis to try to control for what might be confounding factors from one experiment to the other, which you can do in DSeq? If you don't know, that's totally fine too.
Well, I think that's going to be sort of the extension of the project, where I'm seeing like the concordance of data within like respective NGS methods. So I think I'll continue that like during this month and next month.
Excellent. Any other questions? Okay, Sunny has a question. How did you annotate the binding sites to the gene level? Did you select a certain distance, example, within 1 kb from the transcription initiation site?
Right. Within ChIP-seeker, I think the distance to the transcription start site was actually much greater. There's a much greater distance, like both upstream and downstream.
Excellent. Any other last minute question? Feel free to continue the discussion in chat if there are other questions for Amanda. Really well done. And now we'll move on to our next speaker, Alexandria. And Bushra, are you introducing Alexandria?
Hi, I'm here. Well, I will introduce this time.
Perfect.
So this year, yeah, normally around this time I go to like Turkey. I'm on vacation, but this year, of course, there's no vacation. So, yeah, it's my pleasure to introduce Alexandria Westray, and she did a great job. She's actually from Phillips Exeter Academy in New Hampshire. And this is her first year here in the Hillman Academy, and she has been actually very active at her school and the local community there. And she participates, for example, in the biology club, writes for the science magazine, and she's interested in politics and civil actions, and she's part of the Big Brothers and Big Sisters with the elementary school kids in her local town. So actually, I can start my video, sorry. And I forgot to do that. And so we have been actually we developed an EEG-based brain-computer interface to study the distress patterns in the brain in individuals with autism spectrum disorder. So we have been collecting data for the last 2-3 years about this subject, and Alexandria actually helped us with some classification methods and feature selection methods during this summer. So especially like classifying the distress versus non-distress, and like which channels in the on the EEG and which features could be most informative. And she's going to actually present that work today. And I supervised actually the project, but like the special thanks goes to Bushra and Alexandria, and they did actually a great job. Thank you.
Thank you so much for that introduction. Okay, so now I'll share. Okay, can you guys see the presentation?
Yes, looks great.
Okay. All right, so yep, I'm Alexandria, and this summer I worked with Mira Akikaya and Busra Susam, both from the University of Pittsburgh. And so today I'll be presenting on EEG-based BCI classification of distress and non-distress in individuals with autism spectrum disorder. So just some background, individuals with autism spectrum disorder may struggle with emotional regulation. So that means that in stressful and distressing circumstances, they can react with behavior that can injure themselves and others with tantrums or aggression. And there are several electroencephalography components that are directly associated with emotional regulation, including spectral features, meaning frequency domain-based, and temporal features, meaning time domain-based. So something that would really help would be an EEG-based brain-computer interface, which could improve emotional regulation by monitoring these distress components in real time, which would allow for behavioral intervention techniques that could improve lifelong emotional regulation and in that moment, and in the end, increase independence. So the first step in developing an EEG-based BCI is to learn how to effectively classify between distress and non-distress in ASD. So for this project, I had two goals: identify the most informative EEG features for non-distress versus distress classification, and finding the best classifier for recognizing distress. And so all of that is basically for, this goal of having either a stressful scenario as a game or a real life scenario, being able to use EEG measurements for classification of distress or non-distress, and then being able to use an ER-focused intervention to help them in that moment. So identifying features and the best classification method is the foundational step in this. So the experimental setup was that there were 21 individuals with ASD who played an online game based on the effective Posner task. And what that is it's basically a game where you look very straight at the screen at the very center, and something pops up on the right or left very quickly, and the participant has to choose which side it came up on as fast as possible. In total, there were 100 trials, but there were 5 games of 100 trials, but I'm focusing on game 3. So in game 3, there was a deception component where 60% of all responses that were both correct and on time still told the player that they were too slow, resulting in a distress condition. So for each trial, win represented non-distress and lose or too slow both represented distress. While they were playing, EEG data was collected using the DSI-24 system, and players were given $25 just for participating, but were motivated by a bonus of $50 upon collection of a certain number of points. So I analyzed EG data from game 3 because it used the deception component and from 11 participants. And data is collected from 20 channels of the International 1020 system. I collected features from EG data in 3 time ranges: 3 seconds, 1 second, and 400 milliseconds after stimuli. Stimuli in this case being the presentation of a screen saying win, which is non-distress, or too slow and lose, being distress. And then an FIR bandfast filter excluded all irrelevant data from below 0.1 and above 40 hertz. And we used Welch's periodogram method of power spectral estimation to calculate 3 spectral features. So their power of total theta power, and total beta power. Theta power is basically the total power in a frequency range of 4 to 7 hertz, and beta power is the total power in the frequency range of 16 to 20 hertz. We also used temporal features of P200 and P300. That means basically the positive increase or the positive change in EEG over a certain period of time after stimuli. So for P200, it's 200 milliseconds, and P300, it's 300 milliseconds. And so 5 features in each of the 20 channels in each of 3 time ranges resulted in a total of 300 features for potential selection. So here's how we did the feature selection and classification. After calculating the features, obviously, you'd use weighted sequential forward selection. So that means that you start with an empty feature set, and then you add the first feature in your feature vector, and then you calculate, does the classification method do better or worse with that feature? If it does better, then you keep that feature. If it does worse, then you get rid of it. And then you keep adding features one by one until you've tested each of them, and your output subset is only with the features that help and improve the classifier's performance. After selecting features, then I tested 3 different classifiers: support vector machine, linear discriminant analysis, and quadratic discriminant analysis. So support vector machine basically works by trying to find a hyperplane that maximizes the distance between the points of the 2 classes that are closest to each other. And the discriminant analysis and both of those classifiers work to also maximize the distance between the classes, but instead for the mean of those classes. But for linear discriminant analysis, the hyperplane is linear, and for quadratic, it isn't. So here are the results for the feature selection for the channels. So out of all 20, the 10 shown here are selecting the most number of participants data. So on the x-axis, you have the channels, and on the y-axis, you have the number of participants, not the number of features that were in these channels, but just the number of participants that use these channels. So the top 5 are FC, F4, F7, C4, and P3. And this matches with what we already know about the use of frontal channels of F3, FC, and F4 in distress classification, especially in ASD. And then here are the feature selection results for beta power and theta power and all the other features. So once again, on the x-axis, we have the specific feature. And then on the y-axis, we have the number of participants that use that kind of feature. So out of 11 participants, all but 1 used total beta power, which reflects what we already know about beta power's increased amplitude in ASD individuals. And theta power was the next most commonly selected, which we can attribute to greater theta oscillations during reward processing, setting it apart from lose conditions. So now I can get into the classification results. So once we did the feature selection, then we tested 3 different classifiers, and SVM had the greatest overall accuracy at 78.07%, as well as the highest accuracy in distress classification, which was the original goal of this project, to classify distress. So SVM did that remarkably well at 91.04%. And then we have the classification results for LDA and QDA. So LDA and QDA both had pretty similar average accuracies, sensitivities, and specificities to SVM. LDA had the second most accurate distress classification, which is sensitivity, which is specificity at 90.72%. And QDA had the second highest overall accuracy at 69.9%. So in conclusion, the EG-based BCI system was successful in recognizing distress. SVM was the most accurate at distress classification at 91.04%. And the features that were selected of beta power and theta power, as well as the channels of Z and F4, reaffirm what we already know about distress classification. And in the future, I plan to continue to collaborate with my mentors to improve the accuracy of non-distress classification, so the win conditions, as well as to train a multi-layer perceptron. And I would like to thank my mentors, Mira and Busra, because I came into this knowing like very, very little, and they were both very patient with me. And Dr. Boone, Solomon Lifshitz, and the Hillman Cancer Center Academy, University of Pittsburgh, Swanson School of Engineering, and the Doris Duke Charitable Foundation for giving me this opportunity to do everything I've done this summer.
Excellent. Great job, Alexandria. So again, let's unmute, give her a round of applause.
You have to make a new rule every time.
Excellent. Now I'll mute everyone because I know I'm doing that and surprising some people while they're doing other things. Really nice sound. Any questions? Oh, we have one from Sunny. She says, very interesting work. 2 questions. What is the total number of samples you use for the feature selection and model optimization? Second question, there were 21 participants. Was N equal 21? Another question, or I guess that was part of the same question, my fault. Another question is, how did you validate the model? Cross-validation, split of training and test, how did you do this?
Okay, so thank you. So for the total number of samples, it ended up being around 84. I think there were 100 samples in each game, but some of them weren't able to be used for different reasons. So there were 184 samples that were actually used. And then for the number of participants, the experimental setup was with 21, but like some others, I think my computer wasn't like bought and like set up for, classification. So it took a toll on my computer. So I think we shortened it to 11 to make it a bit shorter. So I used 11 participants data. So N equals 11. And then for validation, yep, we use K-fold cross-validation, 5-fold cross-validation.
Excellent. Any other questions? So Alexandria, I have a quick question, and it might be related, but I'm not sure. I noticed when you showed your sensitivities and specificities, you had much higher specificities than sensitivities for most of your models. Do you have an explanation of why, and did you actually select it to be that way? Did you wait to have your model to be more important to be sensitive than specific, or is this just random chance that this happened?
I played around with the weights a lot, and the ones I actually presented were the weights that got the sensitivity for the WID classification the best, because at a point the weights weren't changing the distress classification a lot. It changed by like 0.1%, 0.5%. But what the weights really had an effect on was the WID classification. And so it would either like drag down the accuracy even more because the WID classification would get a lot worse, or what the weight I ended with was the weight that had the WID classification at the best, which meant the average accuracy for most of the participants, the best it could be. So I actually don't know the reason. I'm still wondering. I plan to actually try distress versus baseline classification and non-distress versus baseline, because my mentors also collected that data. So I'm curious to see how it does when compared to not just non-distress versus distress, but to,
Alexandra, why don't you tell that the reason why we have the lower sensitivity for the WIN classification? Because of the deception component that you are analyzing in the game 3.
Right, so we used the deception component in game 3, which meant that there was a higher distress classification for 60%. So because there's more for distress, also I think there's a higher accuracy for distress.
Excellent. Any other last questions? If not, please continue to ask questions if you have any in the chat, and we'll move on to someone Alexandria knows very well. Let's move on to Julian and Wilbert. If you could introduce them, that'd be great.
Hi, yeah, sure, I'll be happy to. Yeah, Julian got in touch with us in the beginning of the summer. Julian Westray, he's a rising junior at the Winchester Thurston School right here in Pittsburgh. and he has been very interested in bioinformatics and biomedical sciences more in general, also in chronic diseases. And so he came to us. We are the coordination center for the Midas network, and a lot of work is being done here on COVID-19. And so Julian was interested to working with us on that, and especially on looking at hospitalization of COVID-19 patients across the country. So he has worked with two of our doctoral students, actually, Jess Salerno and Dasha Pokrodnaya, who've been really very, very helpful in guiding him through the process. And so Julian himself has really shown his great interest in the science and also in learning how to code in R, and has made some great progress there. He's been very interested in learning new things, but also definitely didn't hesitate to share his own thoughts and ideas, and has been working also quite independently to move his research interests forward. So we've all been very impressed with his scientific progress over the summer. It's been a real pleasure to work with him. And we'd like to thank him for joining our team for a few weeks, and also wish him the very best with what undoubtedly will be a very successful career. So please, Julian, go ahead.
Good afternoon, everyone. Thank you so much for coming to listen to my presentation today. My name is Julian Westray, and I attend the Winchester Thurston School in Pittsburgh, Pennsylvania. So this summer, with the help of Dr. Wilbert von Panheis, Jessica Salerno, and Dasha Pokrodnaya, I was lucky enough to have this opportunity to research COVID-19 data. On December 31st, 2019, the first case of severe acute respiratory syndrome, coronavirus 2, also known as SARS-CoV-2, was reported in Wuhan, China. Shortly thereafter, it was dubbed 2019 novel coronavirus 2019 nCoV by the World Health Organization. It was later renamed coronavirus disease 2019, as you all know it, COVID-19. As of August 7th, 2020, COVID-19 has been laboratory confirmed in at least 19 million people globally and is responsible for around 160,000 deaths in the United States. Hospitals have been struggling to provide adequate care to all of their patients because of rising inpatient numbers and the lack of knowledge about the virus. So the novel coronavirus can enter through the body through the eyes, nose, or mouth. Here's a diagram right here, just the virus and how it enters a host cell. After it's entered, it finds healthy cells, particularly healthy lung cells or kidney cells, and by using the cell's ACE2 receptor protein and TMPRSS2 proteins, the virus gains entry to the cells and latches onto them. It dominates and takes control of the cell and eventually kills it, in which case it goes looking for more cells to infect. My hypothesis. The duration of hospitalization for COVID-19 confirmed patients is longer individuals who have been pre-diagnosed with any of these four diseases: diabetes, cardiovascular, liver, or kidney disease, when compared to those who have no such or little diagnosis. Although the novel coronavirus specifically targets lung cells, it impacts other systems, one of which being the cardiovascular system. I believe that infected patients with cardiovascular disease will have a longer duration of hospitalization because the coronavirus can cause vascular inflammation, which leads to the weakening, narrowing, thickening, or scarring of blood vessel walls. Blood flow can also be restricted, and if the patient is suffering from any other cardiovascular disease, additional vascular inflammation can be lethal. The novel coronavirus can also cause myocarditis, which is the inflammation of the myocardium, the heart's muscle, and that can lead to abnormal heart rhythms, also known as arrhythmias. As noted, COVID-19 impacts the cardiovascular system, and the damage on any part of the cardiovascular system can also impact other systems and organs, including the liver and kidneys. Infected patients have experienced increased levels of liver enzymes flowing, including alanine aminotransferase and aminotransferase, as well as new liver damage. Kidney damage has also been reported in COVID-19 patients because kidneys have receptors like the lungs that will allow the virus to attach and infect the cell. When infected, the body releases an excessive amount of cytokines, known as a cytokine storm. Cytokine and an influx of cytokine can damage kidney tissue. And my method, the viruses have been known to be harder to manage in patients with diabetes, with complications from non-fluctuating blood sugars. So my methods, I found verified COVID-19 studies using the Medline database. I recorded data from those studies and exported them as a column separated value file, also in a CSV file, into R. With ggplot, a library in R, I created graphs and plots using the data, and I compared the data and I compared the graphs with all 4 of them, and then I analyzed trends to observe if there were any, my hypothesis was correct. So in this graph, I have liver disease versus duration of hospitalization. I've got each study name and patients and the country, because that'll be important. So with liver disease, it looks like there's a directly proportional relationship. On this outlier right here, Pierce-Williams, that's actually a study conducted on pregnant women with COVID-19. So not only are the doctors caring for the unborn child, they're also caring for the mother, and not only the pregnancy care, but also COVID-19 care, which obviously explains the long duration of hospitalization. So in cardiovascular disease, there is an inversely proportional relationship. Here's the outlier again. So with kidney disease, there's no real relationship because kidney disease wasn't found in many of the studies. There's only 5 points, including the outlier, which obviously is consistent among all the studies as being abnormally high. So look at this going up and going down with the larger percentage of patients with kidney disease. And with diabetes, there's no real relationship. I kind of predicted this because I knew diabetes would be affected like kidney disease and liver disease were. So I knew that there would be a little, there would be something, but there wouldn't be much. I still want to include it in my study just to see, just in case. Why is this happening? My hypothesis only proved correct for liver disease, where there's just a directly proportional relationship. With other ones, cardiovascular disease, kidney disease, and diabetes, there is no relationship for diabetes and kidney disease. However, there's an inversely relationship for cardiovascular disease. So for future research, I believe that I need to conduct research in the same hospital because these are all around the world with many different patients and many different types of disease. Like it wasn't in the studies it listed cardiovascular disease has this percent, liver disease this percent. Instead of saying with this type of liver disease, this patient diagnosed with this, it was just all like one lumped together into one disease. I would like to see how COVID-19 affects patients with different types of cardiovascular disease and different types of liver disease and so on and so forth. I also like to use research that specifically studied comorbidities and the duration of hospitalization because these were often cast aside for the broader topics. I know many of my studies were named clinical outcomes and characteristics of the virus in patients with the virus. However, the comorbidities and the duration of hospitalization together, they were kind of just cast aside for the death, other type of stuff, like death with age, duration of hospitalization with age. And I wanted to study this because it's kind of been thrown to the side. So I think studying this would be interesting because so many people have certain diseases, and I think it would be interesting to show how their disease could correlate to their hospitalization rate if they're to contract the virus. I'd also like to use more recent studies because a few of these studies were conducted in January during the early stage of the virus where not much was known. So I would like to see what it's like in July now in January, how the information about the virus has changed and the knowledge overall in all the countries has changed. So hospitals have more knowledge on what to do with patients, kind of care to give them. So it may turn out that if they know what care to give them, they'll be released earlier, but if they don't know, they'll keep them later just in case because they don't know. Like with the first point, I like to observe the severity of the infection because possibly mild cardiovascular disease is like lumped in with the cardiovascular disease. So it would take away from a point that's high. So maybe severe cardiovascular disease would have direct proportional relationship compared to mild. So I think studying like everything a little bit closer and taking a deeper look would be a lot better. Obviously, it wasn't accessible to me for the studies. So if I were to conduct research in the future, that's what I would do. These are my references, and a big thank you to Dr. Wilbur Panheist, Oscar Salerno, Dasha Fukunaya, Solomon Lifshitz, Dr. David Boone, and the Hillman Cancer Center Academy.
Excellent work. Good job, Julian. Again, I'm going to now unmute everyone. Let's give him a round of applause. Great work. And happy to take any questions that you might have. We have one question from Adrian. He asked, has anyone looked in UPMC patients at these factors in duration of hospitalization?
I have not. I used different studies that were not UPMC related, although that would be really interesting to see because we do have around like 9,000 cases of coronavirus in Pittsburgh. So it could be a good sample size. That would be something I would do in the future because the data is more accessible to me and I could access a lot more because it's right there. So that would be something interesting to do, but I've not done it yet.
Yeah, and we don't have the data from UPMC yet, but we're working on looking at the Western PA network of hospital systems where we could do similar analyses. But I haven't seen studies come out yet about these relationships.
Excellent. Any other questions? Well, if not, Julian, I think you answered all of my questions in your conclusions when I was going to ask about doing it within one hospital system and the differences between countries and the time of diagnoses and what it related to what we knew about the virus at that time. So I think you hit all the points, the questions I was going to ask about potential confounding factors that might influence these correlations that you were looking for. So really nicely done. Okay, and so next we have Eduardo, and I have Sangjin asked to introduce him.
Hi, so I will introduce Eduardo in place of Songjin. It's my pleasure to introduce Eduardo. From my working experience with him, I think Ed is a very smart and independent person. Like when we discuss we need to solve one problem and the next day we'll figure something out on his own or something at least he has already tried. And he's not afraid to ask questions when things got a little confusing. But sometimes when I saw the email, he already replied like, never mind, I got it. So I think, yes, learning and solving problems independently, those are very great qualities for Ed to do this science or basically doing anything else in your life. So let's welcome Eduardo's presentation.
Thank you. One second. Okay, so my research is based upon a gene signature I identified that was associated with metastasis and survival in BRCA patients using a machine learning approach. So in order to understand any of this, we first have to understand why perturbed pathways are significant. As you can see on the left, it is a normal functioning pathway where none of the signal proteins are mutated and the transcription factor then puts out normally regulated genes. And then in a perturbed pathway, as you can see what the arrow is pointing at, this signal protein is mutated, which then leads to the regulated genes by this pathway to be differentially expressed. An example in the real world scenario that we can see is here in this proliferation pathway that the ERB receptor is commonly overexpressed, which can be seen to be an example of a perturbation that leads to cancer. Gene signatures are important to find because they serve as biomarkers. They help create targets for drug discovery. They also help predict clinical outcomes, so they serve as prognostic biomarkers, and they also help serve the future of precision medicine by treating everyone according to their own needs. So our purpose in the experimental approach, we wanted to identify the better machine learning technique for finding gene signatures and also to identify the gene signature in our data with the most significant association to clinical outcomes. And here on the left box, you can see our artificial neural network model. And right below it, we can see BEM algorithms model. And we use both of these techniques to try to extract two matrices of latent factors by gene signatures and latent factors by samples. So what we did first was transform our data to a DEG matrix. So we got our TCGA data, which was log2 normalized, and we inverse log2 it. And we applied the two criteria. If the z-score was greater than 1.645 and the fold change was greater than 2, that value would be 1 if true and 0 if false. And that would spit out our binary matrix. And here we can see the data before being transformed, and then after, where we can easily see the differentially expressed genes. So what we did to identify the genes was we first filtered out our data. So we removed our healthy samples, and also we removed the genes that were not differentially expressed at least once in every 20 samples. And then we determined the number of latent factors through the BEM AIC's function. And then to get the node data, we extracted the edge weights. So we got the first and second edge, but to get the gene signature, we used the second edge. And what we used specifically was the 99th percentile of all values in this matrix to serve as the threshold. And then to correlate the genes with the corresponding clinical data, we got the latent factors by sample matrix, and we got the survival data, and we plotted a Kaplan-Meier curve for each latent factor using disease-specific survival and progression-free interval. At first, we were trying to use Pearson correlation with the tumor stages, but we saw that there was not a significant correlation. So we used this instead, and we picked the gene signature with the most significant clinical outcome. And in our results, we can see BEM outperforms our artificial neural network when looking for gene signatures. As you can see in the box plot, these values are the neg log 2 of the p-value. And we can see that BEM spits out much better p-values than our artificial neural network. And this is due to its ability to pick up subtle patterns with greater variance on factor sizes. And also, we got these p-values through analyzation with Go enrichment. So then we, here I put the Kaplan-Meier curves, and this is for our signature. And as we can see, once the patients that have this gene signature perturbed, once they reach around the 4,300 day mark, the clinical outcomes become really bad. From the progression-free interval going down to a probability of 0, and the disease-specific survival going down to around 37%. And we were wondering why this was the case. And once again, once we analyzed it with Go enrichment, we saw that our gene signature is associated with metastasis. The cell cytoskeleton plays an emerging role in metastasis. And this is because the microtubule structures help mediate cell movement and other cellular events. We can see this in other cancers, such as lung cancer, with the expression of beta tubulin being associated with overall survival and lymphatic metastasis. And then what we can conclude from this research is that BEM is an efficient machine learning technique that can be used to discover gene signatures in RNA-seq data. Our proposed gene signature can be seen to provide poor clinical outcomes in BRCA patients due to its association with metastasis. And our proposed gene signature can serve as a prognostic biomarker in order to better treatment and advance deeper into precision medicine. What I wish we could have done is narrowing down the gene signature, eliminating the genes that do not play a large factor, as our results ended up being in around 470 genes. And I'm grateful for my lab group, Dr. Liu and Lifan, for the priceless mentorship, Dr. Boone and Solomon, the University of Pittsburgh, and the Department of Biomedical Informatics. And yeah.
Excellent. Great job, Eduardo. So again, I'm going to unmute everyone. Let's give him a round of applause. Excellent. And now, are there any questions? Again, feel free to unmute, to type them in the chat, to raise your hand in the participant box. And while we're waiting, Eduardo, I'm going to ask a quick question. So you did a little bit of work to see what your gene signature, what the genes were enriched in with using gene ontology. I'm curious because you specifically noted that in breast cancer, it correlated with poor prognosis. Did you happen to see if that gene signature was strongly associated with triple negative breast cancer, which is known to be to have really poor clinical outcomes, and is also very distinguishable by genes compared to ER positive breast cancer?
Oh, no, we didn't really look into subtypes.
Okay. Yeah, I'm just curious if that, again, knowing that they're very different, I'm wondering if your algorithm's picking up on those very strong differences too. Any other questions for Eduardo? If not, feel free to ask any questions in the chat, and we'll move on to our last talk of this session. Another returning undergraduate, Taylor, who's been with us for 4 or 5 years now, for a long time, but I'll let Meghana, if you want to introduce Taylor.
Certainly. Good afternoon. I'm Meghna River. I'm the Associate Director of the Program Evaluation and Research Unit, which is housed within the University of Pittsburgh School of Pharmacy. And a lot of our work revolves around health policy, some of it touching upon health disparities in different populations. And as a result, Taylor and I spoke a few months ago, and she expressed a lot of interest in the topic. So Taylor has done really good work, a lot of research into the Appalachian region and what the potential impact of COVID could be in this region. Again, limited by data, availability of a lot of data, and she can speak to it. But she's collected data from the CDC's Social Vulnerability Index, from the National Institute of Environmental Health Services Pandemic Vulnerability Index, and other Appalachian Region Commission data to present some of it in a meaningful way that's going to speak about the vulnerabilities and also subsequently the resiliency that's very specific to this region. So that said, Taylor is a senior STAMP scholar at the University of Pittsburgh. She is majoring in communication rhetoric and minor in applied statistics. And she plans to pursue an MPH in epidemiology, and then in the future, hopefully a PhD in the public health sectors. And she is trying to combine her research interests in communication science, as well as picking up some of the pieces in epidemiology and data science to better understand health policy. So let's welcome Taylor Robinson.
Hi, thank you for the introduction. I appreciate it. I'm going to share my screen. Okay. Hi, everyone. I'm Taylor Robinson. I'm a senior at the University of Pittsburgh, And this summer, I had the opportunity to work with Dr. Magana Ruru and the University of Pittsburgh School of Pharmacy. And our project assesses vulnerability in the Appalachian region during the COVID-19 pandemic emergency response. So just to give some background about our focus. So I'll be talking about COVID-19 and community resilience, and then going to the Appalachian region, talking about our methods, and then finally some critique and future applications of the work. COVID-19, also known as SARS-CoV-2, began in Wuhan, China, and has spread globally. There are 19 million cases worldwide and 5 million cases in the United States. We know that there are populations that are at higher risk for serious symptoms, and these populations include older age, those with lung problems, those with chronic conditions like heart disease, diabetes, obesity, or those with weakened immune systems like cancer or HIV. But there are also some social factors that contribute to increased risk of infection and death of COVID-19. And these social factors include things like homelessness, being a part of a racial and ethnic minority group, health access, and also occupation. So knowing that there are social factors that contribute to increased risk of infection and death of COVID-19, we want to focus more on the social vulnerabilities that are specific to COVID-19. So for example, for those that belong to a homeless population or are impoverished, they may have limited housing, which could impact social distancing and the necessary isolation and quarantine policies. These communities are also more likely to face food insecurity. We can just think about the number of people who were hoarding toilet paper and food when the pandemic first began. So we can see how these vulnerabilities exist. Discrimination is another one where racial groups are more likely to face structural racism, which impacts housing, healthcare, and can lead to chronic stress. And then also healthcare access and utilization, where if you have limitations on insurance, or if you rely on public transportation, it may be harder for you to access healthcare. If you face language barriers and cultural differences, that can also impact your utilization of healthcare. And also occupation as well plays a role in vulnerability too, where essential workers have more chances of being exposed and may not have the chance to do remote work or have paid sick leave. We understand where vulnerability exists, and we want to address systemic vulnerabilities. Vulnerability itself has a number of definitions. It could be social conditions that exist prior to a disaster that make a community at higher risk of certain harm. We define it as a property of a system which upon interaction with the hazard produce an outcome, including a disaster, and a disaster being a singular large-scale event. So we want to move from social vulnerability to community resilience. And community resilience is defined as the dynamic properties of a population that involve concepts such as learning, reflection, adaptation. Essentially, resilience is the ability to adapt to a disaster. They are the resources that are available to combat vulnerability in times of crisis. We want to apply these concepts specifically to the Appalachian region. The Appalachian region contains 13 states from upper New York to more southern states in Mississippi and Alabama, and includes 420 counties. 42% of the counties are rural, and they're also northern and southern states. The Appalachian region faces a number of known vulnerabilities. So it suffers from high unemployment, limited transportation, higher rates of heart disease, and low socioeconomic status. Healthcare utilization is pretty low in the area. There are also geographic variations across the region where rural and urban areas face different types of vulnerabilities. So urban areas are somewhat more likely to have resilience factors because they have like a better social infrastructure. But either way, COVID-19 can exacerbate vulnerabilities across the region. And we know that there are gaps in the literature evaluating health disparities and social vulnerability in this region. We want to explore the impact of the COVID-19 pandemic on community resiliency in the Appalachian region by identifying where these vulnerabilities exist. So we retrieved data from the CDC Social Vulnerability Index, the NIEHS Pandemic Vulnerability, and also county health rankings to assess vulnerability in the Appalachian region. We analyzed clusters of counties via agglomerative hierarchical clustering representation in the region by different metrics of vulnerability. And then we explore COVID-19 health policies. So one of the factors that we used to assess vulnerability was the CDC's Social Vulnerability Index called SVI. This uses census data to monitor vulnerability and is ranked on 15 social factors across 4 categories, including socioeconomic status, minority, and like housing and transportation. And a higher SVI score represents higher vulnerability, and it helps to identify areas in need of different policy implementation. The other factor we used to assess vulnerability was the National Institute of Environmental Health Science Pandemic Vulnerability Index. And this is specific to COVID-19 and uses radar cards to score overall pandemic vulnerability by grouping all of this data. So as you can see in the photo on the left, there are different factors that are assessed, like infection rate, health and hospital bids, intervention testing. But the highest relative vulnerability is a score of 1. And this data includes CDC, SVI, and also other factors related to COVID-19. So first, we want to cluster Appalachian counties by vulnerability. So this is our clustering of the Appalachian counties by the Pandemic Vulnerability Index using the unsupervised agglomerative hierarchical clustering. And the image on the left is our produced dendrogram of clusters of pandemic vulnerability counties. And so the images going from left to right on my screen are just like expanded versions of one of the clusters, cluster 3. So it shows all of the counties that are within cluster 3. And so these are Appalachian counties clustered using PVI raw data with Euclidean distance and complete linkage. So once we have the counties, we know that in cluster 1, which is like the cluster at the bottom, is pink in this image. Cluster 1 has 96 counties, and these include states from Alabama, Georgia, Virginia. Cluster 2 has 181 counties and includes more northern states such as Ohio and Pennsylvania, West Virginia. Cluster 3 has 120 counties, most of which are more in the central region like Kentucky, Tennessee, West Virginia. And then finally, cluster 4 has only 14 counties, and all of them are in New York. So even when I change the cluster amounts, this one still remains pretty unique. So cluster 4 is quite different. So when we cluster Appalachian counties, there are some regional associations. So we can see in cluster 1, largely the state count is in the southern region. Cluster 2 is largely in the northern region. Cluster 3 is like in the central region, and cluster 4 is all in New York. And so this image is showing a map of the different types of subregions within the Appalachian area from northern to southern. And so what we can see is that in cluster 1, which is in the southern area, is also within that region in Appalachia. Cluster 2 is in the north, 3 is like in the central, south central region, and cluster 4 is like upper New York, which is a pretty unique cluster within itself. But there are regional associations, or we believe that there are. So now we want to examine vulnerabilities in Appalachian clusters. So these are box plot representation of median values of vulnerability. And what we can see is that when looking at percent diabetes and percent obesity, cluster 3, which is highlighted with the red box and is the blue color, has the highest median vulnerability. We use the notched box plots to demonstrate differences across the clusters and median assessment. So cluster 3 has the highest median vulnerability due to underlying health conditions, while cluster 1 and 2 are pretty similar by percent diabetes and percent obesity. Cluster 3 has the highest median vulnerability and socioeconomic status, and it also has one of the highest vulnerabilities in housing, as well as cluster 4. And cluster 2 and 4 have the highest median case fatality ratios, which defies the other previous trends where it was really cluster 3 and 4. But this is basically representing the case fatality ratios from until June 30th of this year. We also assess COVID-19 policies as well. So this is a table of the Kaiser Family Foundation state COVID-19 health policy actions. And this is based on the Families First Coronavirus Act. And this act helps to ensure equal access to COVID-19 through different coverage policies. So what we can see is that given the if we take out the states specifically that are in the Appalachian region, that there is a variation in the type of policies that are implemented across the region, and that could have an impact on vulnerability. So just to break down the table a little bit more, none of the Appalachian states have actions to waive cost sharing for COVID-19 treatment. And by specific state policies, we can see that 23% of Appalachian states require free vaccine testing, only 23 have paid sick leave, 53% have expanded telehealth services, which is a wider margin, and 15% require a waiver for COVID-19 testing. So even though there are policies implemented to ensure that people have equal access, this is not really enacted as well within the Appalachian area. And to break down the table a little bit more, if we focus specifically on Kentucky, which has some of the largest policies, or at least the most policies out of the Appalachian region that are implemented, we would assume that Kentucky might have the lowest case fatality ratio as well, given that there are more health policies. And so what we can see as well is that might be an association, where in cluster 3, it has the highest count of counties that are in Kentucky, and it also has the lowest cumulative case fatality ratio as of the 30th. So there might be a relationship with implementing health policy and the impact on the spread of the coronavirus. So some of our main takeaways is that case fatality ratios of COVID-19, socioeconomic status, and comorbidity status vary significantly across the Appalachian clusters. And this has implications for policy work and implementation in the region. And so we know that these vulnerabilities exist, so we have to know what's important for each of these regions. Cluster 1 and 2 have similar median diabetes percent and obesity, but different case fatality ratios. So in the news, there's a number of articles about comorbidities and impact of COVID-19, but we can see that these two clusters have similar median values. So why do they have different case fatality ratios? Cluster 3 has the lowest median case fatality ratio, but has the highest vulnerability, diabetes, and obesity percentage. So that could be impacted by policies. And we also know that the Appalachian region has state policies, but they don't currently protect the majority of vulnerable populations. So that may play a role in prevention in Kentucky. Some critique is that COVID-19 is changing daily, and so case fatality ratios may be different in the future. Another limitation is that cluster trends don't reflect individual actions. So although the dendrograms that we created show similarity, they don't necessarily mean significant differences between groups. And then finally, our work addresses just a few aspects of community resilience. So in the future, we hope to analyze other data, including regional differences and perhaps mapping these differences using GIS. There's also possibility to model relationships between vulnerability factors and Appalachian counties to see any type of predictive modeling. That we could create a heat map, or we attempted to do that, but I think that needs to be improved to further explore clusters and factors that are driving clustering. We could explore COVID-19 data past June and understand how vulnerability changes with time of policy implementation. And then finally, we could explore other factors of community resilience. So this is just an image I found of community resilience framework. So although we address like some of the social vulnerabilities, there are other factors like justice, self-advocacy, environmental behaviors and support, and how that plays a role in vulnerability. So just other things that we could look for in the future. Here are my references. Thank you. Thanks to my mentor, Dr. Ruru, and to the Doris Duke internship for sponsoring this research. Thanks to Dr. Boone and Solomon for your support, and to the TAs and Dr. Boone for assisting with ideas and answering questions, which I appreciate. Oh, any questions?
Excellent. Great work, Taylor. I'm now going to unmute everyone. We know the drill. Give her a round of applause. Very nicely done. Any questions for Taylor?
I have a question. So like now that you have this and you know how like different clusters, like the different kinds of the different kinds of actions that certain clusters need, do you plan on like sending this data to like people who work in like public policy to sort of make sure that the different clusters are getting the support they need?
Yes, that's yeah. You touched that pretty much the main goal of it. So we found, or we assessed how Appalachian counties are being clustered by vulnerability, and so the end goal is then to use this data to then guide policy implementation in these regions. So if there's differences across vulnerabilities, how can policy work to target these specific vulnerabilities within the regions? So not just taking the Appalachian region as a overall, but also assessing like individual level factors across the counties.
And have you distributed like your findings to like the people who implement the policy, or it's something of that nature?
I think Dr. Ruru can probably speak to that more, but that might be a long-term goal, probably not at this moment. I think the research has to be like verified. I don't know the process that you go through, but I think that would be one of the goals in the future.
Yeah, and I can add to that. So the Appalachian region is historically a distressed and socioeconomically disadvantaged region. So there are a lot of policies that have driven economic development in the region across the entire belt. But I think getting to the policy level issues, there's much more work that needs to be done in terms of the analysis, drawing specific associations, and then speaking to the resiliency. Because the goal ultimately is to improve the resiliency of these communities so that when they recover from disaster, or as they start to recover from disasters, they are catching up with the rest of the country in other counties, and they're not left far behind. So in order to do that work, there's still more that needs to be done. And certainly one of the goals would be to inform policymakers that these are coming through publications, presentations, and other avenues.
Wow, it's so important. Thank you for answering my question.
Of course.
Excellent. Any last questions? But if not, again, I want to say congratulations. All of you did amazing work this summer. Really nice job on the presentations. Thank you for your persistence. And again, thank you to all the mentors for taking time in these difficult moments that we're living in. So now we are going to take our last break. So again, for those of you who are new to join us and want to stick around, we are going to take a break until 1:45. This this Zoom call will remain open. You can leave, come back in. You can just stop your video and go do something for 10 minutes and come back. But we're going to take a break now, and we'll be back at 1:45 for our last session. So thanks. Okay, so welcome back, everyone. All right, so we are back for our last session. It's been, again, a great day. It's been so much fun to hear everyone's final talks and to celebrate with them. Please stick around this session immediately after. We have 4 talks, and then I'm going to have a very brief conclusion. I will not take very much of your time, but I have a lot of people I want to thank. And again, before we begin, I'm going to thank, again, the students for all of their amazing work this year, and their mentors for giving of your time. Again, I appreciate it so much for giving to these students when you could have easily said no. But with that, we're on our final session. For those of you who are just now joining us and haven't been around, just to explain the logistics of how we've been doing this, and please bear with me, everyone else who have heard me say all of these things 3 or 4 times. The way that this will work is that I will allow the student or the mentor to introduce the student. Then the student will give their 8 to 12 minute talk, followed by a few minutes of questions. Feel free to ask the questions in the chat, to raise your hand in the participant, or to unmute, and we will give the students a few minutes to address their questions. But without further ado, I will turn it over to Ling Zan and to introduce Pablo.
Okay, yeah, can you guys hear me?
Yes.
Okay, okay, this is Liang Zhan from Department ECE. So Mr. Pablo Cohen Parani is a rising senior at Winchester Thurston High School here in Pittsburgh. I believe this is his 2nd year at Hillman Academy. This summer, he worked on data mining on brain networks for Alzheimer's disease. He learned about the brain networks and also different machine learning technique, and also how to visualize brain mapping, and also many other stuff, some of which he will discuss today. He's planning to continue this work onto his senior year and give talks at conference and present at the science fairs. So without further ado, I turn to you, Pablo.
Thank you very much for your introduction. Give me a second to set things up. All right. All right, well, hello everyone. I'm Pablo, and this summer my project was a predictive machine learning model for Alzheimer's disease, detection, severity, and brain node significance. This project was done by myself with the help of Haoteng Tang and my mentor, Dr. Liang Zhan. All right, so let's begin with what is Alzheimer's disease, or AD? Alzheimer's disease is a progressive brain disease that affects memory, behavior, and thinking capabilities to a much greater extent than natural aging does to a human. It is the leading cause of dementia, causing 60 to 80% of the total cases. And essentially what it does is that it destroys neurons and their connections throughout different parts of the brain. And this causes a lot of mass loss in the brain. Here, as you can see from this figure, the healthy brain is quite big, and the advanced Alzheimer's brain has a lot of empty areas. It's quite a bit smaller in the cortex size because of these destroyed neurons. And treatments only slow down the symptoms, and there is no cure yet. So this means that for treatments, the earlier the better, because the earlier we detect it, we can slow down the progress of it, and we can have longer lives for Alzheimer's disease patients. Now, brain networks were the type of data that I used for this project, and they consist of nodes, also known as regions of interest. And these regions of interest are bounded by edges that can be defined using fiber tracking from diffusion MRIs or piercing correlation of bold signals from functioning MRIs. Now, a diffusion MRI is displayed here in this figure. This is a normal MRI, and this is a diffusion MRI. And essentially what a diffusion MRI does is that it provides more contrast in the image, which is useful for both scientists and physicians alike. And now, brain networks networks are capable of capturing the differences in brain dynamics caused by diseases. Now, how do they do this? First, you input a diffusion MRI-derived brain structural network, such as this one here. This is kind of the top-down view, and this is the side view. And then after it goes through this matrix, you output the nodes or brain regions that are affected by Alzheimer's disease, which is this right here. I like this figure a bit better because not only does it show all the different nodes and all the edges, but also the hubs where Alzheimer's is most present. Now, the machine learning technique that I used in this project was support vector machines, which is also known as SVM. It is sort of similar to linear regression in the fact that it involves drawing a line of best fit, or in this case, it's called a hyperplane. And a hyperplane is the line that's drawn the largest distance to the first element of each type of data. So it's kind of a type of line of best fit. But the difference is that in SVM, we will add a third dimension to deal with non-linear decision boundaries. So here's an example of this. Here, you can't really draw a linear line to separate these two different types of data. As you can probably infer, it would have to be a circle. So how do we do this? With a hyperplane, we add a third dimension, the z dimension, which is calculated by doing x squared plus y squared, and that equals z. And here you can see that it's very easy to draw this best hyperplane to separate the two types of data. And then when we subtract this dimension, bring it down to 2 dimensions, you can see that the circle, the best hyperplane, is here, and that's essentially what SVM is. Now, enough about the background. Let's talk a bit about the goals for my project. So my goal was to develop a machine learning model to predict the stage of a potential Alzheimer's disease patient using brain networks. So the different stages we were trying to predict: normal control, also known as NC, is just a normal brain. It's a control brain. MCI, or mild cognitive impairment, is essentially a brain that has been affected by memory loss and thinking loss and everything, and all of these things to a greater extent than normally seen in aging. And then there's full-on Alzheimer's disease too, which is AD, which is the most severe case that we are trying, the most severe stage that we are trying to predict. Now, brain mapping is essentially, we're mapping the nodes that we see. So back in the drawing of the brain network, you saw all those nodes. We're essentially, we're taking all these, this data of the stage, and then we're remapping the nodes. And then deep learning is essentially trying to do the same thing that we did with a machine learning model, but using the more powerful tool such as deep learning. The experimental approach for our model, first step was to extract the features from the data in MATLAB, and then we would load that data and the features into Python and split it into 5 separate parts. That's for cross-validation, of course. And then we will apply a principal component analysis, or PCA, on those 5 separate parts. And PCA compresses the data, which later on in the process, it makes it easier to not only visualize it, but also easier to analyze because it's compressed. Then we will train the model with 4/5 of the split data and use the last fifth to test and the model and obtain the accuracy. Now, using the model we built and our 86 by 86 matrix data, we ran the model 100 times and shuffled all the split data so there were 5 new partitions between every single run. And as you very well may know, this helps prevent data bias. And so we recorded then we recorded the data, and then we graphed it in MATLAB. Now, the procedure for brain nodes significance is a bit different. We would once we had the model done, we would iteratively remove one node from the network and then reevaluate the accuracy with that node taken out. And then based on how important we conclude that this node is, we would then visualize it in MATLAB using the BrainNet package. Now, here is a graph of the accuracy and standard deviation over the first 100 trials for our model. It is an accuracy by frequency graph. On the y-axis, you can see the frequency, so how many times each accuracy appeared in my 100 tests, and the accuracy is on the x-axis. Now, as you can see, we got a mean accuracy of just about 74.53% and a standard deviation of just above 3%. And I think the main takeaway from this graph is that it is quite a precise model given the standard deviation, but the mean, the accuracy could definitely be improved in the future. And now here is a single brain node visualization from multiple angles. Now, so to get a figure like this, the first thing that we did, as it said in the procedure slide, was to remove a node. I just removed a random node, and then I retested for accuracy. Now, for this specific node, it turns out that the accuracy was a lot lower with the node removed than with the node in there and with the full data, which means that this node was key to the differentiation. So key to predicting, it was a key to predicting which stage of Alzheimer's this brain was in. So given the fact that we just concluded that it was key, so it's important, then we can go on to visualize it. And visualizing it is important because we started with MRI scans, so an image, and then we translated that image into numbers, into matrices of data, and now we're bringing it back into image form for the final step. And so here we can see from all different angles, top, bottom, and this is more of a full side image. Now, some conclusions from this project. We built a SVM machine learning model to detect Alzheimer's disease and its stage of progression. As I said before, the model is quite precise, but it could be more accurate compared to what is out there. Especially the deep learning models out there, those are quite accurate. So we're trying to get up to there eventually. Alzheimer's disease is one of the leading causes of death in the world, especially among the elderly population. And since there is no cure, more research on the diagnosis and the treatment of this disease is really crucial to saving more lives in the future. Now, speaking of the future, some future directions that we're looking at. So as you may have noticed, we ran out of time for the deep learning part of our project. We didn't really do it. And this deep learning part involves using deep learning to solve the same problem that our model solved. So predicting whether a brain is an NC brain, an MCI brain, or an AD brain. And then we would compare the deep learning results to the traditional machine learning technique results. So we would compare it to the SVM results. Another future direction was to improve the accuracy of our SVM model with more testing, and not only improve the accuracy, but make sure that it actually is that accurate by doing other statistical tests, such as an ROC curve test and other things. And now finally, to flesh out the brain node feature to make it more comprehensive and easier to use. At this point, it takes quite a bit of time and it's quite clunky to get a node and then visualize it and get to the end. So making it easier to do and more streamlined would definitely be a good thing for us. I would like to thank Dr. Lian Zhang for all his mentorship throughout the summer, all the interesting things he taught me. Mr. Haoteng Tang, who's a PhD student at the University of Pittsburgh, for all his help in coding. Dr. David Boone and Solomon Lifshitz, of course, for organizing this program every year, and especially this year for keeping it online instead of canceling it. That was much appreciated. The DBMI, the Hillman Academy, and the Hillman Cancer Center. And of course, last but not least, the NIH for their generous funding of both the Academy and my research.
Excellent job, Pablo. So again, I'm now, for those of you who are just joining us, at the end of every talk, we unmute and then give them a round of applause. So I'm going to do that now. I'm going to unmute everyone. We can give a round of applause. Excellent work. And now we have time for a few questions, if anyone has any questions.
If you did have time to finish your the deep learning model, do you think you would have gotten much better results than you did?
I think we would have gotten better results, much better, I'm not sure, because deep learning takes a lot of time, especially someone like me who has absolutely no idea what I'm doing in the space. I don't know where to start. So I think it would have been a very preliminary, a very like surface deep learning model that hasn't been trained very much because we wouldn't have time. I think it would have definitely been better, but not fantastic like some of the ones that you see in other papers.
Excellent. Any other questions? So Pablo, I know we talked a little bit about this yesterday, but I can't remember if you said you've looked at, if you just like superficially looked at the data at all. You measured accuracy. I know in your conclusions, you said you're going to look into area under ROC curves and sensitivity specificity. Did you happen to look at any of the predictions that were made and see if it was better one way or the other? Would you, if it said it was a normal or a quote unquote healthy brain, was it very good at predicting if it was a healthy brain? Do you know if there were more type 1, type 2 errors, or maybe you don't know at all? I don't know if you've kind of looked.
Yeah, I don't really know at all. That's definitely something that would be easy to find out though, from run some commands.
Cool. Any other questions? If not, or if you still have some, please continue to ask in the chat and he can continue to answer there. But next up we have Anissa, and I'll have Steve introduce her.
Hi folks, I'm Steve Abramowitz. I'm from the Department of Bioengineering at the University of Pittsburgh, and it was absolutely my pleasure to host Anissa this summer. I got to know Anissa, I got to know Aubrey and Ace, some siblings, so you know, it was really quite a pleasure to meet with her on a weekly basis, and even more often than that on some occasions. Anissa is a rising junior. She is a member of the Pittsburgh Science and Technology Academy. I see Dr. Kitchington is on the call. So hello, Edwina. Thank you for supporting this student. Anissa's fantastic. She's been Science Student of the Year, honor roll for quite a few semesters. She's participated in Investing Now and the Gene Team. She also has participated in cross country and the Interact Club. She's a member of the Black Student Union, Stand Together for Change, and a student ambassador for SciTech. This summer, we had her doing some segmentation on some data that we received from what's called the Korean Visible Human Project, which was you know, a huge set of data, and she was able to process it and really deliver using a couple of different pieces of software, some useful information that I think will help to guide clinicians and certainly our clinicians that work with us very much moving forward. So with that, I will turn it over to Anissa and just want to just share how proud I am of what she's accomplished this summer.
Thanks.
There it is.
Yeah, my computer, I just updated it, so it was acting weird.
Perfect.
So my presentation is going to be about creating a 3D representation of normal female pelvic anatomy using MRI and CT data. So first, for some background, I want to talk about pelvic floor dysfunction in women specifically. Pelvic floor dysfunction is characterized as the weakening of the pelvic floor muscle, which you can see here in figure 1, so I'll explain the anatomy. In the pelvis, there are these organs located there. So there's the bladder, urethra, the uterus, the vagina, the rectum, and then the pelvic floor muscle is here, which wraps around them. So this is the anterior of the body, the pubic bone, and then the posterior, this is the spine here. And there's a few different types of female pelvic floor disorders, such as urinary dysfunction, anal dysfunction, pelvic organ prolapse, and sexual dysfunction and chronic pain. And it's important for me to talk about these things because they impact more than 50% of Paris women. So women that have given birth once or multiple times. So a lot of women are affected by some form of dysfunction, and more than 11% of these women eventually undergo surgery. So a lot of them, these impact a lot of women's lives. And so the visualization of the pelvic anatomy is really important, but it's hard to visualize because taking a 2D image and making it 3D is hard, especially when there's not a lot of detail, like in a lot of scans. And so this could lead to surgical errors, especially with a lot of trainees, because they'll be familiar with women that already have some form of pelvic dysfunction, so they won't know what normal anatomy looks like. So for example, here in figure 2, and I'll explain later, but the way that this pelvic floor muscle is set up, it doesn't make any sense, honestly. And so with those surgical errors, they also lead to morbidity. So these women are going into surgery to fix these complications and then coming out with more problems. So, like I said before, the clinical scans that are used are typically of women that already have these anatomical defects. So over here is an example of an MRI scan of a woman that already has pelvic organ prolapse. So the rectum here is pushing into the vagina. It's sort of hard to see. And, few trainees understand what's actually going on in the body when there is no dysfunction. So some background about the basis I'm using for my model is the visible Korean human data. So they took a lot of highly detailed imaging, specifically CT and MRI images of cadavers. So some information about the MRI and CT is that the MRI uses a 3 Tesla magnet, and it had a slice thickness of 1 millimeter. And that's important to note because a lot of clinical scans are typically 3 to 5 millimeters. So there's a lot more detail in the MRI than there usually are in clinical scans. And the CT also has a slice thickness of 1 millimeter. And some information about the cadaver that I had. She was a 43-year-old female, and she had a height of 152 centimeters and a weight of 54 kilograms. So my goal here was to create an accurate 3D representation of normal female pelvic anatomy based on the segmentations from the Korean Visible Human data set, and then render that anatomy in a 3D rendering software called Blender. So for my methodology, I started with segmentations, and to do that, I used a software called Seg3D, which was made by the University of Utah. And so basically this allows you to combine the hundreds of images that these MRI and CT scans take and put them together to make basically a sort of like this. This is an example of one of the layers, but it puts them all together. And so first I used a CT scan for the pelvis. And so CT scan is just basically an x-ray. And I used something called the threshold tool, which automatically allowed it to highlight all the bone and differentiate that from all the body tissue. And then after I did that in a separate project, I segmented the organs. This is an example of what I did down there. And I combined T1 and T2 weighted MRI images. So T1 highlights fat in the body, and T2 highlights liquid in the body. So here you can see this is the bladder, and it allowed me to find where the wall was and differentiate that from the liquid inside. And so after I did that, after I made these segmentations, I made an ISO surface in Seg3D still, which basically combined all of the segmentations I did in each image and sort of made like a 3D type model. But the issue there was that it was made of voxels, so these small cubes, which made the anatomy really rough and not realistic. So I imported those ISO surfaces into Blender, and then I had to smooth the model. So in Blender, there's a lot of these things called modifiers, which basically allow you to manipulate whatever you input into it. So I was able to smooth everything out, and then I added procedural materials onto the model. So instead of painting directly onto the surface of all the organs and everything, I use these things called nodes. And it's an example of one of the ones I used here, where I could get the textures and the color that I wanted without having to do anything sort of manually. And so after I did that, I rendered the whole model by itself, and this is what it looks like. So I included some still images just to make the anatomy more clear. So in the earlier pictures I showed, the pelvic floor muscle connected to the pubic bone, which I found in my MRI images wasn't the case. The clitoris isn't a pelvic organ, but I included to show how the pelvic floor muscle goes from the coccyx of the spine, so this part down here, and then wraps around that. And then here's the rest of the organ anatomy. So in conclusion, I was able to successfully create a 3D representation of normal female pelvic anatomy from the Korean Visible Human data set. I was able to create scientific renderings using Blender, and I developed a workflow to generate these renderings from medical images that can be used in the future. And my next steps are going to be to further refine the materials in the models. I had complete creative control over it, and what they looked like was up to me, but the nodes and everything could be a lot better. I have to prepare the muscle model that I made for use as a template in a graduate student work in Dr. Obama's lab. And then I'm going to demonstrate this model to other clinicians for their input. So for acknowledgements, I want to thank my mentor, Dr. Abramowitz, for all the time he took to help me with this super busy schedule, and Megan Rootsong for helping me identify the pelvic floor, which is like the hardest part of the whole model, as well as David and Solomon just for being really supportive throughout this whole process and accepting me into the program, as well as the National Institute of Health for funding us. So, yeah.
Excellent job, Anissa. So again, let's unmute and give Anissa the round of applause she so deserves. Excellent work. Are there any questions?
Other students who presented said that some of the computational work has taken like a toll on their computers. Like, did it take a long time to render the model?
I mean, trying to figure out what software to use took us a second because originally I was going to use this other one called, what's it called, Houdini. And it's sort of similar to Blender, but my laptop just would not accept it. So yeah, it did take some time. And if I had a different computer, it probably would have worked a lot faster. But
It's an excellent question, and something that we thought about too whenever doing this remotely. It's another one of the challenges because we can't just like hand you something when something's not working, right? But good question. Any other questions? So Anissa, I'm still curious, how many images did you have to curate to build that model?
in total, I think there were 512 images. I don't know if Dr. Abramowitz like exactly remembers. I don't know how it works exactly. But I sort of use like different planes. So I used the sagittal and coronal planes. Just switching between those to like accurately identify different anatomies. So I don't know how many it took, honestly. It was me flipping back and forth between a bunch of them until I like figured it out.
Yeah, that's excellent. And I'm curious, so this is the model of one woman. Can the process and the pipeline that you built now make it more readily accessible to do this to other women's pelvic anatomy? Can you use some of the same processes you built, or is there so much manual segmentation that it's still going to take a very, very long time to do this again with other people?
You could do it with other people, but it's still gonna have to be manual just because everyone's anatomy is slightly different, and trying to make a computer like do that is sort of hard. So like, yeah, my workflow could be used for like other segmentations, but it just has to be like, I guess, tailored to however the anatomy works, wherever you can identify.
Yeah, excellent. And we have a question in the chat. So Dr. K asked, how did you feel about learning the computational skills and applying that to biological problems?
At first, I was also nervous. I did do a little bit of coding, and it just wasn't my thing. But I really enjoyed like the whole, this whole modeling aspect, and it's definitely something I want to use more in the future. So it was pretty great.
Excellent. Any other final questions? Okay, if not, or if there are, please continue to ask questions in chat. And that will move on to our next talk from Angelina and Jenna. And Rich, if you'd like to introduce them. I think Rich is here. I saw him earlier.
I'm not sure if he's here.
Oh, okay. Well, I will, oh, what is this? Oh, I am in on Chris Boyce. Okay, so he is here. Solomon, can you give audio permissions to Chris Boyce?
Yeah, 1 second. There we go.
All right, so now you should be able to unmute, Rich.
Can you hear me okay?
Yes.
Okay. Hold on a sec. Sorry for that. We were trying to get a different computer working. Of course, it's not my usual account. So it's good. So anyways, that's good. So anyways, I wanted to introduce Angelina and Jenna. Both of them have been amazing students to work with over this summer. Angelina is a rising senior at Western Pennsylvania School for the Deaf, and has a strong interest in both computer science and biology. And one of the cool things about our field is we get to put the things together. And Jenna has an interest in lab technology, and we didn't touch much on biology, but we did bring someone in who has a PhD in that area, and we were able to talk about some stuff related to cancer research, which I think was of interest to her. Both the students, of course, are excellent students in their high schools, and they have very bright futures in the deaf community and also for going off to college. So it's a real pleasure to work with them, and super proud of the work that they did this summer. And so that's my introduction, and looking forward to their presentation. Thank you.
Thank you for that introduction, Rich. So I'll go ahead and share my screen now. Okay, so yes, hello, my name is Jenna, and I work together with Angelina this summer. And our project that we've been working on, it's been ongoing for a few years. So Angelina and I both were repairing some issues with the tools. And let me get to the next slide. So both of us go to the Western Pennsylvania School for the Deaf, and our mentor is Dr. Richard Boyce. So some of the issues that we were working on were developing a novel tool to help deaf high school students learn biology. And I can't seem to advance to the next slide. I'm not sure. Oh, there it goes. There we go. Okay, so some background about the project. So deaf and hard of hearing students are underrepresented in STEM fields, including bioinformatics. We do have deaf scientists. They're out there, but there aren't enough of them. So we need to recruit more deaf scientists. It's only about 7% of deaf or hard of hearing people who work in STEM field that have received a master's degree or a PhD. And that is compared with 12.1% of hearing students. So one thing you should know is that deaf people are very visual learners. They learn by using charts, diagrams, images, and we use our eyes and our hands to understand concepts in the world. And we use American Sign Language to communicate with other deaf people, which is a very visual language. So some barriers that occur frequently in deaf and hard of hearing students are due to a lack of self-advocacy, a lack of accommodations, and a lack of a support system. Our hypothesis with this project was that if we combine information technology that interactively links material that's highly visual to specific terms and phrases in the written material, that it'll allow the deaf students to have better topic comprehension acquisition. So I'm going to summarize some of our prior work and some of the prior work in the project and what it looks like. So the top left image that you see is a Wikipedia enhanced page, and there are yellow and blue terms that are highlighted on that page. So for example, if you were to click on the word outcome, it's a blue highlighted word, meaning that it's a general English term. Yellow highlighted words are scientific terms. So if I were to click on the word outcome, it would display a bottom row that would show on the left images, and it would have a definition of that term. It would have pictures related to that term, and it would automatically play a slideshow of those images. And then on the right half of the screen, you would see a video of either me or Angelina signing. And what we would be signing is we would be finger spelling the word, whatever the sign for that word is, and giving a definition in ASL of what that means so that the deaf person can understand the topic and the term that's being described. On the bottom right of the slide, you'll see that there's a YouTube video. And this is an example. If you click play on the video, it'll show us signing, and automatically captions will pop up on the bottom. And it will automatically, whenever you click on a highlighted word, such as chain, for example, within the captions, it will automatically play that. And the video automatically pauses itself, so you don't have to do anything. And you can, look on another part of the screen to see the definition, the pictures, and all the descriptions. So the methods, our goal was to recruit 15 to 20 deaf or hard of hearing participants whose first language is ASL. And we're planning to conduct a pilot within subject user study of the tool. And this is the layout. I'll show you the design here. And so what you see with the red box, it means that they had help from the tool. If there's no red coloring in the box, it means they did not receive help from the tool. So there were 3 subjects, and they're given within the first row. Subject 1, the test 1 didn't receive help from the tool, then they did, and then they didn't. So it was randomized within the subjects within the test to make sure that we could look at the data and separate out from the results whether or not the tool was helpful or not. So that's the plan for the future. Our work this past summer was that we were to prepare for this pilot study. So we made all new ASL videos with the tool help to making sure that it was, use standard procedure for length, appearance, level of detail. And then we tested the tool with the target users to see, was it usable? What feedback did we get from those users? And now I'm going to let Angelina take it over and describe her part.
Should I share my screen or just go with this? Okay,
you can go ahead,
one
Angelina.
moment. Okay. All right, are we able to see it?
Yep.
Okay, I'm going to go ahead and begin. So Angelina, I'm Angelina, as Jenna just introduced me. So the results of what we've been doing and working on the slide here. So we had 82 videos of biology words, 42 of the terms were scientific words, and 40 of them are English words. And I'm going to show you a little bit what this looks like. And what we did, we worked on to make the toolbox more accessible. So you can see here the toolbox on the right-hand side. There's yellow and blue. Yellow is for scientific terms and blue is for English ones. So as you can see on the toolbox on the right, there's basically you would be able to have the option to hide, or you could add, for example, you could hide 2 of the terms, or you could hide the scientific phrases or the English phrases. It's like a parent-child concept that basically you're able to pick for exactly what you want to view on the screen. So the progress right now with the IRB protocol. So it's still pending. We're finalizing the consent and the promotional documents and waiting to hear from them. And we've tested it with 2 users so far. We did the full study procedure. So that was a pre-test, tool assistance, and the post-test to make sure everything works smoothly. So we did pre-test, the tool assistance, and the post-test. And then we fixed some issues as we went through. For example, some of the videos weren't playing because permission issues. We had minor technical issues selecting words and viewing some of the figures. We added some new features to the website to make it easier to use, and we changed the researcher script to address issues, the ones that require training. So now we finally have a stable tool, and we're ready to do the study once we get IRB approval. And what we've learned, what did we learn? We learned a lot of different biology terms. Again, so it's 82 videos, so that was a lot. And it was a user-centered software design. So it was iterative requirements, design, and testing we went through. So the conclusion, we were really glad to have this opportunity to help the deaf community. And for the future work, we want to continue the program, and we're hoping that IRB will approve us and we can do testing with other deaf people. And we plan to continue to study after Hillman Academy if it is possible to continue this work. For the acknowledgements, of course, there's a lot of people to thank. So first, the 2 students who actually originally set this project up last year, Haley Snodgrass and Jasmine Blank. And to the interpreters, we want to thank them for being here, to doing all the interpreting. We have the Max, who worked on as a programmer for the tool and the web page, and we had 2 deaf people who actually attended some of our meetings to give us some advice and work. And first was Dr. Lauren Farovich and Kenneth Dahan, and of course, we want to thank Solomon and David for helping us in the program. Thank you.
Excellent job. So thank you. And instead of unmuting this time, I would like for us to give the visual a round of applause for Jenna. So it's like this. So congratulations. Wonderful. So we have a couple of questions already in the chat. So the first one, can you explain the tool again? Is it specifically for Wikipedia or to implement across other biology-related platforms?
So it could be basically any website. We just used Wikipedia for the study, but it could be other ones as well. It could be a variety.
Yes.
Excellent. Any other questions? Can you, I'll ask a question. Can you explain how you decided which terms to do? So you said you did over 80 videos, which is amazing. How did you decide which biology terms to record videos for?
So yeah, I know there are common terms that, really anybody might have a hard time. One second, let me fix this. That might have a difficult terminology. So we decided that those would be the high priority ones that definitely need to be clarified. So we made videos for those ones. So really, in general, that's how that worked.
Very cool. Any other questions? Okay, we have another question from Taylor. What do you think was the most challenging term or concept?
Oh gosh, honestly, there were so many words that I was just like, wow, I didn't even think of that word. So, I had to do some research myself, or I had to ask my mentor or to figure out that term, because honestly, there were so many of them. I found a lot of vocabulary terms that were really challenging in there. So yeah.
Yeah, I agree.
Excellent work. Any last minute questions? If not, again, congratulations. Well done. If there are more questions, please continue asking them through the chat feature. And we are going to now hand it over for our last talk of the day from Neil. And Doug, if you'd want to introduce him. Solomon, can you make sure he's unmuted? I see he's not a co-host.
Okay.
Perfect.
I think I am now. So
yeah.
Last but not least, Neil was an outstanding student this year. He's going into his sophomore year at Connecticut, double majoring in English and physiology and neurobiology, an 8-year program that's a combined bachelor's and MD, and has a minor in pure mathematics. He has a lot of varied interests, from writing and poetry to music and piano and volunteering with the Coast Guard, and even has a patent pending on a spacer attachment for asthma inhalers. So he really needs to get busy and do something with his life here. He's was great to work with. Neil took over some work that I've had a few students work on with model complexity of neural networks. And I don't think he really had much of a background in regression, much less neural networks, and really spent the first few weeks just self-teaching himself. I assumed he was going to do that, and then I was going to have to explain it to him. But the meaning to explain it to him part never came. He figured it out and has done some really interesting simulations. So I'll let him take it from here.
Hello, everyone. Thank you for the wonderful introduction, Professor. Let me share my screen. Can you all see that?
Yes, perfect.
Perfect. So I am a third-year returning student to the Hillman Academy, and this summer I worked in the lab of Dr. Lansdell in biostatistics, and we looked at neural networks and how we could estimate their model complexity. So a little bit of background into artificial neural networks. ANNs are designed as computing systems designed to simulate associative memory in the brain. That is, they're indiscriminate in recognizing complex patterns, which means that if you feed them a large set of data, which the human mind may see bias in, an artificial neural network typically will not, and they'll be able to recognize patterns that we wouldn't otherwise see. So ANNs have applications in quantum chemistry, game theory, statistics, medical diagnosis, engineering, basically everything you can think of that has large data sets. And I'm going to be focusing on statistics. One of the biggest and most efficient types of artificial neural network is what's called a backpropagation neural network, or a backprop network. And what it does is it adjusts the connection weights to compensate for learning error, which means as you feed it more data, it becomes more and more accurate. One method that a neural network can do this is something called stochastic gradient descent. Mechanics aren't very important, but as you can see, you start with the maxima and you're trying to approach a minima. The minima is the area of highest precision and lowest bias for your network, and you take little steps going down past the gradient part. Another type of neural network is the feed-forward neural network, and that's exactly what it sounds like. It's a network where information only passes in one direction, contrasted to a recurrent neural network where data is fed back and forth through the same nodes. So if you put a feed-forward neural network and a backprop neural network together, you get what's known as a multi-layer backpropagation perceptron network. That's a lot of big words for what's saying it's an efficient network that information flows in one direction, which becomes more accurate as you feed it more data. These commonly use a sigmoid activation function, which is just the logistic equation. And one big tenet of backprop networks, especially multi-layer ones, is their model complexity, which is the number of features in a predictive model and the class of the model. So here we have an example of a neural network. It has two hidden layers in the middle. It has its input layer and a single output layer. And really, we can classify the model complexity of this network, but we lack the tools to do it efficiently, and it's not really clear how to apply that to other fields where it's important to choose a type of network. So The model complexity of ANNs remains difficult to estimate. And in the past few decades, there have been quite a few methods proposed, but all suffer to some degree. You can represent a neural network as a statistical regression. And so here we see that for a network with 3 inputs to a single hidden layer and 1 output, the important part of this is that it's a single hidden layer. You have the regression model given by y hat and the connections and hidden units given by h sub j. If we extend this to 2 hidden layers, we get, again, the model looks like this, we get the following regression. And this is important because it shows that how deeply related artificial neural networks and statistics are, so that we can efficiently model the complexity of these neural networks. So my project and our objective was to estimate the null complexity of neural networks for dichotomous Bernoulli outcomes using different types of inputs. What this means is we take a neural network, we feed it a variety of data ranging that's either binary or continuous, either 0 and 1 or anywhere from -1 to 1, and we let it predict binary outcomes. Bernoulli outcome is like, for example, the presence or the absence of a disease, and we see how well that matches up to our estimation for model complexity. So what we do is we have 500 to 2,000 observations, and then we take 200 to 500 simulations. And what this looks like is 200 to 500 arrays, and each matrix, 200 to 500 matrices, and each matrix has 500 to 2,000 observations. So for example, if it's binary, we have 2,000 times 500 data points. And each matrix gets fed through the neural network. And then that happens 500 times, 1 time for each matrix. And we get a set of predictions. After we have the set of predictions, we calculate their deviance using this equation. And under the null hypothesis, since this is all randomly generated data, we hypothesize that the deviance is equal to the mean chi-squared value of the degrees of freedom of the neural network, and that the variance is twice the deviance. So hence, the most important part of this is that we can say that the mean deviance of the network measures the null complexity of the model. That is, if we have a neural network and we're trying to estimate its model complexity, we feed it this number of data points and we calculate its deviance, and that measures its complexity. So I began by replicating some previously published data which looked at binary inputs and binary outputs and a single hidden layer. And what we found was that all of our mean to variance ratio was almost exactly 2, which is exactly as you predicted. And the way we can confirm that our data is correct is by looking at what's known as a quantile-quartile comparison plot. And that maps our data points to the chi-squared distribution. And that tells us how closely our neural network fit our data and produce predictions. As we can see, the prediction is quite good. Next, I repeated this with 2 hidden layers, extending what had been done previously. Again, this is the highest level of network that I looked at. It has 2 hidden layers, each with 10 units and 10 inputs and a single output. This is with binary inputs. So this is an example of the data. And as we see with binary inputs, all of our data still has an approximate double variance to the mean. And so our original hypothesis of the degrees of freedom being calculated by the mean holds. So this actually continued up to 3 hidden layers, and I could test it with 4 hidden layers, but I figured out the hypothesis would be similar. So next I moved to continuous inputs. And here's just a QQ plot showing the correlation. Again, we can see that it's pretty much exact. So with the continuous input, instead of just feeding our neural network zeros and ones, we feed it a combination of values from -1 to 1 with a mean of 0 and a standard deviation of 1. And so here's where the data gets really interesting. Here are 2 hidden layers and 3 hidden layers. And what we see is that the mean to variance ratio is no longer 2. In fact, it doesn't follow any recognizable pattern. And our mean and variance increase quite a large amount. This last row is the highest simulation I did. quite a few hours to run, actually. And we get a variance that's approximately 100 times that of the mean. So what does this mean? Well, I plotted the QQ plus to see if this was accurate. And sure enough, for most of the simulations, it was. However, if we look at the QQ plots for data with 2 hidden units and 10 hidden units per hidden layer, we actually get an S-shaped curve like this, which means that our prediction is not holding to the chi-square distribution. This is with 2 inputs. So, of course, I replicated this with 3 inputs. And it's the exact same pattern, which leads me to believe that when the first hidden layer has significantly fewer hidden units than the second hidden layer, our data no longer follows the chi-square distribution. That was quite interesting because it meant that our original hypothesis wasn't actually predictive of a chi-square distribution predicting the mean degrees of freedom and model complexity. So what this means is that the chi-square distribution does adequately approximate the degrees of freedom for binary inputs, but this is not the same for continuous inputs. There are patterns in how the continuous inputs produce mean variance, but it's not easily estimatable in the same way that binary inputs are. What this also shows us is that degrees of freedom for any type of neural network is dependent on number of inputs, hidden layers, and hidden units. I messed with a whole bunch of parameters in our networks, including the threshold and learning rate and the type of optimizer used, and they all seem to follow a similar pattern. So what this helps us do is understand that there's a new outlook on statistically analyzing neural network efficacy. So if you have a really, really large complex data set, you can effectively choose a neural network based on its model complexity by looking at how many parameters you have and then comparing it to your type of data and looking at the model complexity of the network that would best fit it. I've also been replicating a lot of this work in Python using TensorFlow and Keras, and that has been really incremental in validating our findings. I'm still working on that. And the next step is to examine these results with more parameters, different types of inputs, and apply it to other real-world situations where we can effectively determine the model complexity of a network for a set of complex data. I'd like to acknowledge my PI, Dr. Lanzittel, as well as the Hillman Academy team, especially Dr. Boone and Solomon. I've worked with you all for quite a few years now, and every year it's always been one of the most favorite parts of my year. I'd like to acknowledge the DBMI and the Hillman Cancer Center, and of course my family, who I have been living with since college has shut down. Thank you. Any questions?
Excellent work, Neil. So again, I'm going to unmute everyone for the last time to give a round of applause to Neil. Excellent. Any questions? So Neil, I'll ask one while we're waiting to see if anyone types anything in chat. So at the very end, you mentioned wanting to apply this to real world data sets. Do you have a type of data set in mind that you would like to do this? Genomics data, clinical data, something else completely entirely that has nothing to do with healthcare? You're muted, Neil. I unmuted you. Go ahead.
Thank you. Going off of all the research that our lab does, I would say that it would be most applicable for clinical data sets where we're looking at one, Bernoulli outcomes, so the presence or the absence of a disease, or a yes or no type qualification of a data set. I think those would be most efficient. Of course, we could increase the number of outputs in our neural networks and then see if we could apply this to other types of data sets as well. The computational power needed would be pretty intensive. But yeah, I think those single Bernoulli outcome data sets.
Excellent. Any other questions for Neil?
I might have seen something in the chat.
Well, it's just Sam, and I don't understand it. What kind of pizza? This must be an inside joke. I have no idea what that means.
Oh, it's the graphic on the side of the slide.
Ah, I see. Okay, if there are no other questions, Neil, if you don't mind, stop sharing screen. I'm going to now conclude with the program. For those of you who have been here in the past, this is normally a much bigger ordeal. It takes a long time. This will not take a long time because we are not in person, but that doesn't mean it's not as important. So let me share my screen. And are you seeing the proper screen? Not like the note screen? Awesome. And let me just try to move some things out of the way. So it's, again, I think all of the students need to be absolutely commended for the work they've done and the mentors.
It's still showing the note screen, I think.
Oh, it is. Okay, perfect. Let me swap screens. Thanks, Solomon. Is it the right screen now?
Yeah.
All right, perfect.
That's the right one.
So thank you all for being here, for the families that have come, the community members, the teachers, everyone who is here. Thank you so much for coming. I wish we were still in person and we were celebrating. And in addition to these talks, we typically would take over the entire bottom floor of the Hillman Cancer Center and have a large poster session. And just the buzz that's happening during that is unbelievable and always the highlight of my year. I wish we were doing that. I wish that we would have our typical graduation ceremony where I could hand you your certificates in person, but we will still be mailing them to you. We are also creating the abstract books, and you don't have them right now, but we will mail those to you so that you still have these physical manifestations of the work that you've done with us that we hope help you as you go forward applying to colleges and doing other things. But again, thank you for being here. This is our 12th year of doing this program. We started with 5 students. Last year we had close to 80, with including the undergrads. And this year is the first time we've ever done this virtually. And it was planned kind of at the last minute and on the fly. And so thank you to all of the students, mentors, for bearing with us as we were trying to figure out how best to do this program too. Likewise, almost all of you are supported by the NCI through the YES program and also the Doris Duke program. And I want to thank them, even though they're not on this call, for being willing to be flexible with our program as well and allowing us to still continue to do this, even though it's not quite exactly what we anticipated doing, but also to the Hillman Cancer Center and to Pitt for hosting students and being supportive. But most of all, I just want to say, as this advances, congratulations. Congratulations to all the students. You did an unbelievable job. I am seriously fully blown away with the amount of work that you were able to do in 7 weeks, having never met your mentors. Some of you have never done computational work, maybe not even were interested in computational work coming into it. And to give these presentations in less than 7 weeks is unbelievable. So I want to say thank you. And even though we can't shake hands and receive your certificate, I just want to say thank you and congratulations to Pablo, Sam, and Kieran, who were here last year. That's why they're highlighted in red. To the first year students, Erica, Eduardo, Marcus, Amira, Alexandria, Angelina, Jenna, Amanda, Oscar, Trayvon, Anissa, Julian, Jonathan, for all of the work that you were able to do under pretty bad circumstances that we're all in. So it's absolutely amazing. Also to the students who I've known for a long time now, Yash and Taylor and Yaser and Neil, for coming back not only to do research, and they've been here for longer, for 9 or 10 weeks, but also to serve as leaders a bit this year and to help in designing the program. And Taylor especially hosted a couple of sessions and gave a talk herself to students. And I'm just, again, I'm blessed to know all of you. Welcome to the family. And, we don't have our normal picture that we take of all of us. We just have all of these random squares. And I don't even have everyone on this image, but we now definitely have everyone. And I always say that the future is bright, but even in dismal times, being around all of you this entire summer and seeing the work you've done have given me such great hope for the future of research and of our country. So thank you so much for all that you've done. I wish we were in person so I could see your posters and do other things, but we'll still make posters because we're not finished. I guess this is kind of the cool thing is of being a smaller group is that you're all one cohort that belongs to the YES program now. So this is not your ending. You know, this was our proposal. It's slightly different now. We are just finishing research experience for year one for most of you. Some of you, it's the end of your second research experience or your undergraduates coming back. But we now want to work with you throughout the school year. And now we are very familiar with how to do this virtually, since that's all that you know. So we will continue to work together to translate the work that you've done into hopefully science fairs or college applications and scholarship opportunities. We're here for you. We talked about this last week, but for the family members and mentors involved, they have some requirements that they have to do. And if I can get these slides to advance, one of the requirements that all the students have to do is enter into at least 2 science fairs, competitions, abstracts to symposium. I've asked every student to submit to the American Medical Informatics Association high school scholars program, which is part of their annual conference. So the largest conference in the world dealing with medical informatics. All of you have projects, I think, that are relevant enough to this. We've designed the abstracts that you've already written to fit their format, likewise with their talks. Over the next week, I will take a look at all of them that you've submitted and try to give them back to you with some feedback. You can talk to your mentors and refine it as well. That abstract is due September 9th. So please, everyone enter into that and then enter into at least one other fair. And this could be anything. We've talked about this already. If you're in Pittsburgh, the Pittsburgh Regional Science and Engineering Fair will happen virtually this year. The first deadline is November 29th, so we still have some time. The International Science Fair, we have time as well. We've had students who have qualified for all of these things in the past. For all of you that are rising seniors in high school, I highly encourage you to enter into the Regeneron Student Talent Search. That's going to require some work, so we need to get moving to do that. It's due November 12th and requires a paper. But we've had students over the last several years who have won money by entering into this. And again, I think all of you have a big advantage this year, as I know less research has been done by high school students across the country. So I really encourage you to enter into all of these things. So choose at least 2. Likewise, every year Pitt usually has a science symposium. And it's called Science, and then whatever the year is. I have not heard yet if they're going to move this virtually, if they're going to be doing this in the fall, but if they do, you'll hear from me. And if it's virtual, this again is the advantage is that everyone can do it, not just students who are very close by to campus. But this is always a great time. Oftentimes Nobel laureates or future Nobel laureates give talks at this, and you can present with it. The other really cool thing, we are really, really hoping, as I'm sure all of you are, that by next summer, we can be around people again, and we can all be together. You can be in different research labs, and because you were with us this year, all of you are guaranteed admission to our program next summer as well. So come back, especially those of you who are more interested in doing wet lab work. We will place you with wet lab work. So please come back. This is the point of our YES program, is to be engaged with you for 2 years, including 2 research summers. So please come back. As you progress into undergraduate, there are opportunities for you to work with us through the Doris Duke undergraduate internship. And you've met these students several times, so you can ask them about any of these questions. There are also what are called research education, undergraduate education opportunities. You've met Tech Bio students several times this summer, but there are other ones here at the University of Pittsburgh as well. So whether computational or not, there are opportunities to come back and do research with us at Pitt. As you move on and graduate, I hope, and this is what I did, I never thought that I had a chance at a research career whenever I was at a very small high school in southwestern Pennsylvania, until I had no idea what research was, until a teacher told me to do a program like this. But after doing it, also gave me confidence to look out or seek out opportunities at school whenever I went to undergraduate. And I hope you'll do that too. And then likewise, whenever we come back into meeting in person, we do have a residential program. We hire 3 to 4 residential advisors who we will also connect with mentors each year to live with the high school students. and to be leaders and to control them. And so please, we reach out to our alumni for that opportunity as well. So keep that in mind as we go forward. And I should say, I put this here to remind me again, we've seen lots of younger brothers and sisters and siblings in the background. We really hope that you encourage them to come to us as well. We've had some, some history of that in the past, and we hope that we've built your trust and you've had a good enough time that you could recommend this to your siblings too. And so congratulations again to the students. Thank you so much. And now I'm going to try to thank the people who made this possible. Starting with my bosses, Dr. Ferris, who's the director of the Hillman Cancer Center, highly supportive of this program and always mentors students. Of course, he does wet lab work and is also a clinician and is running the Cancer Center, so he did not mentor anyone this year, but very supportive of the program. I thank him so much for giving me the opportunity to do this with the Hillman Cancer Center name. Likewise, my direct boss, Mike Bisich, who's the chair of my department, is so supportive of this type of work and allows this to be a big portion of my job, which I couldn't do otherwise without his support. And he does everything to support the people he works with. So thanks to Mike at the DBMI and everyone at the DBMI. And likewise, everyone else at the Hillman Cancer Center. Chris Beckenest is the chair of the Education and Training Committee at the Hillman Cancer Center and helps to run this program. He and I talk about the program. We had a lot of conversations when we were talking about what we should do for this year. Cancel, not cancel, everything that everyone is having conversations now about school and sports and everything coming in the fall. We were having those conversations in March through April as we were deciding what to do. Other senior research and administrative leadership at the Hillman Cancer Center who encouraged us to do different things and really advised us. So thanks to all of them too. I want to give an extra special thank you to Solomon, who does so much for this program. He, again, is always juggling 4,000 balls from, I don't know, maybe February through the end of August, and then only slightly less than that the rest of the year. But from managing our website, controlling our application, doing all of the communicating with students, with parents of applicants, as addition to the people who are here, coordinating with our HR department, our Disability Resource Service department, our general counsel's office, our office volunteer, basically every department that exists in the bureaucracy of the University of Pittsburgh. Solomon knows someone there because we have to, and he knows how to get things done. He, again, more than anyone, we discussed how to do this virtually, if we should do this virtually. And he had so many great ideas, like the use of Discord, of bringing in different speakers to talk about empathy and to talk about different things. So, Solomon is truly a co-leader of this program. It does so much work and puts a lot of time and dedication into it. So thanks so much, Solomon, for all you do. You're amazing at what you do. I also would like to thank Dr. Ayub, who I haven't seen today, but he is the associate director of the program, always very busy around this time. And again, is the other main person that I've spent hours and hours and hours talking to throughout the year, but especially about how to do this virtually and remotely and the importance of doing so. So thanks to Joe, if you listen to this later. I also want to thank all of the site heads. So for those of you, this is your first year, you maybe have read about this when you did our application. Typically, all of the students are divided across 6 different sites. Each of those sites have faculty members and postdocs who help to run those sites, almost like 6 separate programs. And so at Cancer Biology, this is Deb Galson and Laura Pascal. Tech Drive X, you guys have all met Andy Duncan, who gave a talk and has been in and out today, Peter Alexander. I run the Cosby site with lots of support at the DBMI. Computational Biology is Dr. Ayub, David Coase, who's mentored students this year, Tim, who also always met their students. At our Immunology and Cancer Immunotherapy, Doctors Delgoff and Bruno, who do an amazing job there. At the Women's Cancer Research Center, we've all saw Steffi today, Jenny, and others who run the program there. We work all year to do this, so even though you didn't see a whole lot of them this year, they still prepared as if we were going to be in person and did a ton of work. They also all recruited some of the great speakers that we had through the year and have given great recommendations. And they're just such fantastic colleagues who give of their time, and so delighted to know them and to work with them. And of course, I've said this 1,000 times today, but this program is absolutely nothing without the mentors. That mentorship and the authentic research that you get to do with your mentors is what this whole program is about. It does not exist. It has no purpose without it. So the mentors this year are, almost all of them I know so well, and again, are just amazing colleagues, amazing people, that even when their labs are struggling, their labs are shut down for a few months, the graduate students and postdocs are worried about their futures and getting work done, they still found time to give to the students. And I mean, obviously, the students all did very well because they're driven, but they had such great help from their mentors. And I cannot thank you enough. Again, Lian Zhang, David and Paul, Jishnu, Natasha and Casey, Songjun, Uma and Will, Murat, Bushra, Rich, Steffi, Jenny, Asiri, Shasong, Sanghoon, Roger, Steve, Wilbert, Giannis, Matt, Chakra, Megana, Sophia, Doug, and all the others. Again, I don't have quite all of your pictures, but most of them here. Just amazing people. Thank you so much for all that you do. Again, you're the lifeblood of this program, so thank you so much. I also want to thank the Doris Duke undergrads again, because again, you guys were absolutely phenomenal this year. Taylor for running the public health panel, and Neil and Yash answering questions on Discord, coding questions that others had. Yaser, I know, even having conversations with people for a long time and just turning into leaders to help run this program. You know, thank you so much for the work that you've done. Again, not only as a student doing research, but also as leaders in helping this program. So thank you so much. I want to give a special thank you to all of our lecturers and presenters. So many of them, I think, gave really nice talks. A lot of times I try to limit the number of talks. This year we were worried, without being in person, that we should have more content. And we had so many people reach out to us, and I think, again, gave freely of their time. Samantha, and Dr. Sen, and Paula, Andreas, Abby, and Dara, Andy, and Kara, Nilgan, and Mike, and Roger, Samantha, David, Lynn, Paul, Duca, Teresa, Timothy, Davin, Mark, Aisha, Inigo. It was all, I mean, it's just unbelievable. Again, from machine learning lectures to breast cancer lectures to imaging, we had a wide variety of talks from all across our university. Again, thank you so much for giving of your time. I'm going to give another special thank you to our teaching assistants. Again, something new this year, as you know, we were worried that a lot of times students learn from one another. They're, especially doing computational work, they're around each other all day in a room on normal days, and they ask each other questions, help each other out. And we tried to replicate that a bit this year, but we tried to supplement it because they can't walk down the hallway and talk to a grad student, postdoc, or mentor if they have a small problem. So we created these Discord channels, and these teaching assistants, coding assistants answered questions all the time, you know, synchronously and asynchronously throughout the course of the entire summer. They were so helpful. So again, you'll see some of these names several times, to both some of our current Doris Duke students, Yaser, Neil, Taylor, and Yash, past Doris Duke students, Kirthana, Shania, David, other past alumni like Ritwick, and then just people at the DBMI, Paul, and other people that Tech Bio Mentoring Committee that we were able to put together. And I want to give a special shout out to Ritwick, who is an alumni of the program from, I don't know, many years ago now, maybe 2014, 2013. He has graduated from Pitt, is now a researcher at CMU in Silicon Valley, who again called me in April and said, we have to do something with these students, and how can I help? And he was the one who was able to reach out to a lot of people I didn't know at CMU to help with a lot of the coding. So thanks to him. And the same thing with past Doris Duke students, again, reached out to me, set up a call for the same thing. They just said that, you know, we think this is important. How can we help? How can we make sure that this program goes on? even a virtual setting. And all of them give such amazing ideas that we've implemented. So thanks to all of you. And of course, this doesn't happen in a vacuum. Mentioned how Solomon is connected to everything in the university. This is true, but we work with great administrators, administrative assistants throughout many different departments in HR, fiscal, DRS, our interpreters who are amazing, our IT people who, again, we get computers in and we're asking them to set them up and so we can mail them out within a day or two to those who needed them. So thanks to all of you for all of the work you've done to let this program happen too. I want to give another special thanks to our community. We primarily work out of the University of Pittsburgh and the Pittsburgh community. And there's just some amazing teachers who allow me into their classroom to talk to their students and do different work with them, who directly recruit for us. We saw some of these, like Dr. K, who was on the call today, and some others. So thank you so much for, again, trusting us to work with the young learners that you work with. Other community members who would meet me in a church on some random Tuesday evening to talk about the program and how we can get more people involved. Fame, Michelle was here today, and others who, again, just bring us such amazing students and partner with us in some outreach events too. The Homeless Children's Education Fund, and Chris, who was here today, Pittsburgh Promise, Terry and Empower House, our intermediate unit here in Allegheny County, Pittsburgh Public Schools, Kashif Henderson, the Black Student Union at Pittsburgh Public Schools, who again, all just allowed me to talk to them several times to get some amazing students. Remake Learning, other Pitt pre-college programs, Investing Now, Gene Team, Camp BioE, the Tech Learning Initiative. There's just some great programs that are here for the young learners in our community, and I'm blessed to have them all work with me. And then, of course, thank you, a special thank you to the parents, the guardians, the families for, again, trusting us to work with your students all summer, for them to do lots and lots of hours with us and to do it from behind a screen. We really appreciate the trust that you've given us. I hope that we connect again. This is, again, the other part that I'm missing this year by doing this virtually is I'm not getting to meet you now, but hopefully in the near future, we'll be able to all meet and we can have some community events and get to meet all of you in person. Please reach out to me if you have any feedback. I'd love to hear from you of your perspective from a family member as well. So thank you so much. And I mentioned this early, but I'm going to mention it again because this is so critical. We are only able to do what we do because we've been fortunate enough to have federal and foundational support. The vast majority of our money is now coming from the NIH, specifically the National Cancer Institute and the YES program, through the R25, through an expiring NIH Cure grant, who was able to pay almost all of your stipends this year and allow us to buy laptops if we need it and to do anything that we needed. The NIH has been absolutely amazing. So your federal tax dollars are being put to good use, hopefully. Likewise, the Doris Duke Foundation, who supported us for the last almost decade of providing students the opportunity, both high school and undergrads. So that's been wonderful. In the Jack Kent Cooke Foundation, who've supported some of our students. The UPMC, both the Parking and Security Office, Diversity and Inclusion Office support us every year. Lenzer and Coach, typically we would hire a bus every year to get students around. We didn't do that this year, but they still support us every year by giving us a significant discount. The Hillman Cancer Center, that helps provide some of our salaries and again, supports us in so many ways and gives both Pitt, the University of Pittsburgh, and the Hillman Cancer Center allow our mentors time to work with students. And that, again, our program would be nothing without them. The Hillman family, the Stan Marks Foundation, also financial supporters of us. Grateful parents and patients have supported us in the past. So thanks, a special thanks to all of our funders. And so that is it. A special call to action because, and again, this is kind of the cool part. You guys are all part of the ESO program. So we expect you now to keep in touch. So we're not just asking you to, we expect you to. I sent you a survey late last night There might be one other or two other like really short surveys. This one's long because it asks about every aspect of the program. It's going to be really important that you do that for us this year in case we do have to do this virtually, so we can learn how to do it better. So please complete your surveys and do that today while it's still fresh in your mind. Submit to AMIA. We have about a month. I'll be emailing all of you or working with you through Google Classroom to work on your papers. We will schedule calls, so we'll set up some doodle polls. And we won't have a lot of calls. We might have one a month. We might have one every 2 months of synchronous work. Most of it will happen is just you continuing to work on your research by writing it up, by presenting it, and us helping with that asynchronously, which we've talked about before. So please answer our emails, engage on Google Classroom. We'll keep Discord open too. So continue to engage in there, get to know each other better too. And then the last thing that I'll ask you as we move forward is to spread the word. If you enjoyed our program, if you think others will benefit from it, please let your friends know, right? So what we found is this nice feedback circle of where really great students allow us to recruit great mentors. And of course, because we have great mentors, we're able to recruit great students. And so we want to keep this going for as long as possible. And with that, I want to thank you. It's been a wonderful summer. Always my favorite part of the summer, favorite time of the year is this, and really today. And you know, again, I'm incredibly proud of all of you. You did such amazing work. So thank you. I look forward to keep working with you. And that's it. So take some well-deserved time off as you're deciding what to do for the school in the fall. Take some time off, and you'll hear from us very soon though. So thanks to everyone. Take care, everyone.
Thank you, Debbie.
Thanks.
Thank you.
Thank you so much.
Thank you.
Thank you.
Thank you.